Okay, it's time to get this started and welcome everybody. We're here today to honor and remember the life of Ralph Yount and how he impacted all of us at WSU and elsewhere. This picture of Ralph is one of my favorites I dug out of the archives. And I don't know what year it was, but it doesn't matter. He looked the same all the time. I want to point out to everybody that there's a picture book over there on the table from about 1996 maybe through 2004. And if you're not in there, the faculty you remember might be there. I came here, Ralph hired me in 1976. I don't know if this works, the next one. And I hadn't changed a whole lot either. So next year is my 50th year here. I retired 2 years ago, and but it's almost simultaneously my grant was renewed for 5 more years, so I'm chained to the bench for 5 more years. It's actually great. And when I came in '76, Keith Dunker had been here, I think, for about a year. By the next slide. There's Keith when I came back then, and then about 2 years later, it was followed by Mick Smerdon. Next slide. And Mick hasn't changed much either. The unit that we were hired into was the program in biochemistry and biophysics. And that later became a department. After my interview, in 1976, I chose to come here primarily because of my interaction with Bud Ryan and Ralph Yount. And they were 2 people that served as my mentors for all the time I was here. The teaching and graduate training in the unit was characterized by, I fought for the word, but the word I came up with was rigor. And Ralph was the one who was behind that rigor, and he set the standard. The unit was relatively small, but most of the faculty were funded, and we had enough graduate students so that everybody had some. Several noticeable differences between now, as I'm still associated with the graduate program, and then come to mind. First of all, the lights in Fulmer Hall were on all night, and sometimes on weekends. That doesn't happen anymore. The society is different, and our graduate students are unionized. Of the incoming class of graduate students, less than 50% made it through the program. That was in the first 10 years I was there. That's really not the case anymore either. We sent faculty out to other colleges and universities to recruit, and that sort of has fallen by the wayside now. And one thing I remember is, and some of you may remember this fondly, we went through several iterations of qualifying exams in order to get you to the prelims. And the one I remember the most is we had 2 questions, and you had to pass 6 of these qualifying exams, if I remember right. And they were made up by 2 of the faculty members. And it wasn't long before we realized that nobody was passing them. And then we found that the only ones that knew the answers were the 2 people that were writing them. So we have gone through iteration after iteration of getting to the prelim, and I think now that most of us use a application for a grant or something as a starting point. Our courses in biochemistry and in proteins and enzymes were the weed out classes for the university. And Ralph taught most of both of those. The program grew out of chemistry and remained intimate with chemistry during those first years. If I can have the next slide, please. This is a picture of Ralph at the Yount Symposium, which was a biotech symposium and a symposium to honor his 65th birthday. And the ex-students there are Boyd Haley, Richard Barclay, and Gerald Carlson. Gerald Carlson's here. And the keynote speaker in this symposium was Dan Koshland, who Ralph did some postdoctoral work with. And remember that as a very fun time. Now, in 1999, after a study by 3 National Academy members who found that our biology departments were really small and that we would benefit by forming larger units, we formed the School of Molecular Biosciences. And by combining the departments of biochemistry, cell biology, and microbiology. After that, we kept the 3 undergraduate degrees, so they kept the 3 degrees in biochemistry, microbiology, and cell biology and genetics. But we fused the PhD degrees into a PhD in molecular biosciences. We currently have 41 students in that program. And the interesting thing is it's more than 80% female, which is consistent with the national trends and what's happening at the university at the undergraduate level. In the undergraduate degrees, we have 190 undergraduate biochemistry majors, which is, I think, fantastic because the university itself has lost 5,000 undergraduate students in the last 5 years. So we've kept increasing them. But Ralph did not like this change because the emphasis was moved away from chemical biology to more physiological biochemistry. We had numerous discussions, nice discussions, about the merits of this change, and it can be argued from several points of view. I found a quote from Ralph's writing that summarized his concerns. Quote, "I liken chemistry to a giant oak tree in which the periphery is growing rapidly, but the center is not. Chemistry remains a central science, with the periphery being the interface of chemistry with material, environmental, and biological sciences." And that was Ralph's strong commitment. Ultimately, at the request of the provost, the School of Molecular Biosciences moved out to the vet school, and that further removed us from chemistry. Ralph was a mentor to me in 2 different areas. One was obviously research. His emphasis on rigor and doing it at WSU made an impression on me. I'm sure Ralph had other opportunities to move to another institution, and whenever I got itchy feet, he would sit me down and carefully explain the error of my thinking. And more importantly, as an administrator, he listened, and he was compassionate. The older I got, the more I adopted Ralph's love of Pullman and the area around here. One of our colleagues used to say, Pullman is at the eastern edge of the habitable portion of the United States. And the longer I'm here, the more I believe in that. The rest of this I want to read from a letter that I wrote nominating him for an honorary degree in 2018. And this was at the request of Lisa Gloss, who's sitting over here. His research involved the examination of how myosin, the contractile protein in muscles, can change shape and ultimately lead to the movement of the organism. The significance of his can change shape and ultimately lead to movement of the, The significance of the research was described in a letter nominating him for membership to the National Academy of Science. It stated, his research was central to validating and interpreting the atomic resolution model of myosin, and the array of nucleotide derivatives and analogs he developed has revealed a new understanding of molecular motors and has been utilized in thousands of studies revealing new insights. This research had important implications for people with muscular dystrophy and other muscle motor diseases. Perhaps the most significant indicator of the value of his research is the fact that his major grant was continuously funded without interruption for 43 years. He was president of the Biophysical Society from 94 to 95, president of the Federation of American Society for Experimental Biology, that's FOSSIB, from 97 to 98, and during his tenure as FOSSIB chairman, he testified to Congress on funding of research. At a time in his career when most individuals are looking towards retirement, he took on the chairmanship of the Department of Chemistry. He did so because of his concern for the future of that department and what its demise would mean to Washington State. In his tenure as chair of chemistry, he increased morale, hired several new successful faculty members, and gave the unit a sense of hope and a direction that were previously lacking. This type of giving of himself to the institution characterized Professor Yount. In scientific circles, he was Mr. WSU. His concerns for this university and his quest to maintain excellence have been hallmarks of his career. When many other faculty members gave up on Washington State University, Professor Yount worked all the harder to make it better. His contributions to Washington State University did not stop at his research or his service or his excellent research record. He has done the little things that make it a very caring and human institution. When the father of one of our secretaries died, Ralph gave her a plane ticket so she could attend the funeral. When one of our assistant professors had a baby, Ralph picked up her teaching load. When the receptionist to the Department of Chemistry retired, he gave her and her husband a trip to Hawaii. He volunteered to be an undergraduate student advisor so he could better understand the current undergraduates. He did not do these things for self-promotion, but he did them because he cared about people, and especially the people at Washington State University. When President Lin Randall started the Eminent Faculty Award in 2001 as the highest award for total faculty excellence over an extended period, Ralph Yount was the first recipient. Ralph, to quote President Lin Randall at the time, he said, "Ralph Yount is what a faculty member should be. He questions every decision, is a tireless and brilliant researcher, loves to teach, and is a superb colleague. I do not think anyone was surprised that Ralph was selected to be the first recipient of this award." Again, quoting President Rollins, "It's people like Ralph that make me proud to be at WSU." If I have the last picture, please. This is, again, at that same symposium, and a picture of Val and Ralph. And that's the end of my remarks. I think the next person up is Ross Dalbey. He's a professor, Department of Chemistry and Biochemistry at Ohio State University. He was one of Ralph's students. And the title of his is Breaking and Entering, Two Tales from a Career in Protein Trafficking. Ross?
It's a pleasure being here at this celebration of Ralph's legacy. So I was a student 1978 to 1983. And if I could have the first slide. The next slide. So this is a picture of Ralph. It's his 65th birthday. We had a reunion, a lab reunion here at Washington State University. Many of you guys were here, so on the left side. And on the right is a picture of Ralph with Dan Koshland. Dan Koshland was his postdoctoral advisor, very close. Can I have the next slide? So I met Ralph for the first time when I visited Pullman, you know, as part of selecting graduate schools. And I had talked to him on the phone before that time. You know, he convinced me to come here and visit. And I met Ralph and Jim Magnuson. And somehow, so Ralph was a really good salesman. Somehow he figured out that I liked classical music and that my stereo was not good enough. So he gave me a fellowship so I could buy a better stereo system. And so that it worked. I came here because of that. I mean, he really knew what people wanted. Could I have the next slide? Or the next bullet? So let me tell you a little bit about Ralph's lab. So it was a very interesting, very energetic, exciting environment. The way I could describe it, we had a lot of characters and bright students, really bright students. For instance, we had Jim Wells, we had Bill Perkins, we had Phil Busby, and a number of other people. And then during the summer, we had Kei Nakamaya, who was a professor at Gonzaga, visit. We really had a great time in the lab. And really, we had fun day and night. It was crazy at times, but really fun day and night. And the lab provided excellent training in protein chemistry and biophysical techniques, as well as the use of nucleotide analogs to probe the active site of myosin. So if you look at the next bullet. So Ralph, as a mentor, you know, he's a very caring mentor, very supportive, and he really tried to help everyone succeed in the lab, right, so that they could work at their best. He had very high expectations, right? He would always tell you the truth. And by the way, the truth was not always what you wanted to hear. You know, he could be very, very hard, but I thought in the end that was, you know, you needed to hear the truth. I think Jim will probably have some more to talk to you about that. The next, and so what he meant to me, he really inspired me to go into academics. And I must say the career was really exciting and fulfilling. You know, I really love my career and going into to be able to do academics. So I could have the next slide, please. So what I'm going to do is just tell you about two discoveries in my lab that were really inspired by Ralph. So let's see if I have a pointer here. Is that a pointer? So I have to go to the screen? Well, anyhow, if you look at the bottom, so these proteins are made in a precursor form with a signal peptide. And then they are moved across the membrane through the SEC-YEG complex, and that forms a membrane-spanning aqueous channel. And what I was interested in was the protease, signal peptidase, would actually cleaved off the signal peptide. So we knew at the time that the signal peptidase, you could not inhibit it by inhibitors against the 4 classes of proteases. So I wanted to characterize its mechanism. So on the next slide, we used the brute force method. And what we did, we found that there was 2 residues that were absolutely essential for activity, serine 90 and lysine 145. So we thought it might use a serine-lysine dyad, very different than the standard prototypic serine protease that used the triad, a serine, histidine, aspartic acid triad. So if you turn to the next slide, now this is a particular experiment that's completely inspired by Ralph's lab. So what we did was we changed, so we did site-directed chemical modification to explore the essential lysine. So we changed the lysine to a cysteine in a no cysteine background. So this enzyme was inactive. So if you look at that left panel, it's hard without a pointer, but you see that band, and what we did is that we treated the cysteine 145 mutant with bromoethylamine, and what you do, you create a thiolysine analog, right? And if you look at the bottom left panel, you see no treatment with bromoethylamine. It's completely inactive. You see no cleavage even at the highest concentration of the mutant. But if you treat it with 2-bromoethylamine, then you see recovery, and then you see cleavage at the highest concentration, then 1 to 10, and then a little bit at 1 to 100. So this again supported the idea, as you can see on the right side in B, it was a serine lysine dyad and not the prototypic serine protease. So if you look at the next slide, we finally solved the structure of the catalytic domain of signal peptidase. If you turn the next slide, please. And this was done by my student Mark Tetzo, and actually worked with Natalie Stranatka to solve the structure. And if you look there, serine 90 and lysine 145 are within hydrogen bonding distance. And when we solved this, we did it with the serine 90 covalently attached to a beta-lactam inhibitor. So it really showed it was a serine lysine dyad enzyme. And this is more common than you might expect. So if you look at the next slide, you see now, if we're looking back, you see there's a number of proteases that use this mechanism. So it's somewhat common, but still it's a lot, it's not as common as the prototypic serine protease. So anyhow, this project was really inspired by Ralph. And actually, of all my projects, this is what I really enjoyed the most, being trained in protein chemistry. So if you turn to, the next slide, next discovery from my lab. So we explored, we discovered that YID-C actually plays a function in membrane protein insertion. So if you look at YID-C, it's an insertase. So it actually inserts membrane proteins into the bilayer, right? It could do so independently on the right side, or it can work in conjunction with the SEC-YEG complex to insert proteins in the membrane. For instance, on the left side, you see a 5-spanning membrane protein. So I just briefly tell you how we discovered this on the next slide. Next slide. And what we did, this was done by Jim Samuelson, and we constructed, by the way, this is all in E. coli, Lin Randall's favorite organism, also mine. And so what we did, we constructed a YID-C inducible strain under the control of arabinose. So if you add arabinose, you express YID-C, but if you grow it in glucose, YID-C is not expressed. So if you look at the plates, the top one, right, you see growth, but on the left side, if you grow it in glucose, there's no growth whatsoever. So it's a central protein. And then what we did, we tested whether this protein on the right schematic bottom, if it inserts, it gets cleaved by signal peptidase. So if you have YID-C, you express it, you add arabinose, you see it's cleaved to the smaller coat form. And then if you add protease, external protease, you see the coat is completely degraded. You see that? But if you do it under glucose conditions where YID-C is depleted, then you see the higher form, right? And then if you add protease, it's completely resistant. So this protein accumulated in the cytosol when YID-C was not present. So YID-C is essential for its insertion. So this is sort of an important discovery that we made in 2000. And then we showed by a technique that Ralph would really like. Next slide. This is done by Minyoung Chen in the lab. And we did it in vitro. We used an in vitro cell-free system, transcription translation system, and then we truncated the messenger RNA. If you do this in the presence of inverted membrane vesicles, you can actually create these insertion intermediates. So if you look at that, the ribosome there, then you have YID-C, and you have the substrate trap. Then what we did is that we introduced a photoactivatable lysine, a diserine analog, a photoactivatable analog at specific positions within this membrane protein substrate at threonine 20, 27, and leucine 34. And then if you look, in the cell-free system, the only thing radioactive was our membrane protein substrate. You can see the black material at the bottom of the gel. And then if you shine light, plus UV light, you see that top band in each of the positions. And then if you use YID-C antibody on the right panel, you see it's immunoprecipitated. So that's actually YID-C that's cross-linked to the radioactive membrane protein that's been trapped in a membrane insertion. So the next slide just shows what YID-C looks like. And this was work by Osama Nareki. He solved it, he's at the University of Tokyo. And it's not like SEC-Y-EG has a membrane spanning aqueous channel. YID-C actually has this hydrophilic cavity with a strictly conserved arginine right in the center of the cavity. And what it does, that hydrophilic cavity, it actually causes membrane thinning, and also the hydrophilic domains of membrane proteins that have to cross the bilayer go through that hydrophilic cavity. So what YID-C does, it decreases the membrane crossing distance, so for the energy barrier for the hydrophilic domains of membrane proteins to cross. Can you click that bottom right? That's actually a movie, see if it works. Yeah, click that thing there. So we did molecular dynamics simulation. This is work done with Steve White. And you can actually see the waters actually penetrating that hydrophilic cavity up into the center of the membrane. Okay, and then the next slide. So YID-C is found in bacteria, mitochondria, and chloroplasts. So in mitochondria, it's in the inner membrane. So it inserts all the proteins that are encoded by the mitochondrial genome. There's like, in yeast there's 7, in humans there's 15. So all those proteins are inserted from the matrix into the inner membrane. It uses the homolog OXA1. And then also chloroplast has a homolog in the thylakoid involved in insertion. And then if you look at the right, there's been an explosion recently, the last 7 years. There's been like 8 or 9 Nature papers are all on these membrane protein complexes that have the YID-C homolog. And the human TMCO has been shown to play an essential role for inserting multi-spanning membrane proteins. So YID-C actually plays a critical role for membrane protein insertion for these multi-spanning proteins. And then I'm going to end on the last slide. Oh, this one here. So how Washington State University prepared me for my career and success. Go to the next bullet. So training in the program was excellent. Now, so you heard from Dr. Griswold about how challenging the program was. The CUME systems. The CUME systems were hard, were very hard. And I might have been one of the people you were talking about that really struggled getting through. And I think that one of the hardest was with Papa John Kladdakooty. I still remember his CUMEs were very tough. And then we also had we had to defend 2 proposals, right? 2 proposals, one on our research area. That was great because, you know, we'd learn about our research project. But another one on another topic, right? And what Ralph said was very useful. He said, Ross, do it on a topic that you want to do a postdoc on. So I said to myself, geez, what am I interested in? And then about this time, I decided, you know, Lin Randall actually was working on protein export, and I got my proposal you might forget, but I actually did it on the signal hypothesis, if you remember. So anyhow, that was wonderful, great training. And then if you go to the next slide, so Washington State had great teachers, RGY, by the way, we call Ralph RGY. Bruce McFadden, Jim Magnuson, some of you guys will know these names. Mick Smerdon, of course, you've heard of. Michael Griswold, Keith Dunker. I remember meeting Keith when I visited. Very enthusiastic, infectious personality. I really enjoyed that. So we had great teachers, and then Jerry Hazelbauer and Lin Randall. And I want to just mention briefly Lin Randall, because Lin Randall, maybe she doesn't realize, but it had a big impact on my career. So in terms of postdocing, you know, I had a couple choices, but Lin suggested Bill Wickner. And I ended up going to Bill Wickner, and I really fell in love with how proteins insert in the membrane, and it became my career. So I want to thank Lin for everything she has done for me. So your teachers can do really important things for you. And if you go to the next slide, I just want to end here. And I want to say something about Valerie, because Valerie actually had a big impact on Ralph. And I learned this later when I visited Ralph in 2000. I think it was around 2000. We went to a movie saying, what was it? I forget exactly the movie. But Valerie, it turns out we had this common hobby. We love classical music, and she knows everything about opera. And I actually am sort of a Handel opera person. And the other important thing about Valerie, she's a lifelong cat lover. So that's, you know, my family, we've never had less than 4 cats. So there's a lot of similarity. So I want to thank Valerie, who, by the way, was trained as a chemist, you know, for everything she did in the background. Okay, thank you.
Quickly, I was trying to remember what faculty members are still around from the ones when I came here, and I remembered Ron Brosmer is still in town. For those of you who remember Ron, I ran into him the other day, he's 91 at this point in time. And I tried to get a hold of him, but I couldn't to come to this. So the next speaker is Jerry Carlson, professor of biochemistry and molecular biology at the University of Kansas Medical Center, and the title is Yet Another Large Enzyme from Rabbit Loin. Jerry.
Thank you. I came down to Pullman from Spokane. Is there a good place to stand? From Spokane in the fall of 1964 as a freshman undergraduate, trying to figure out what to do, and pretty clueless. Fortunately, the first 2 people I met in the first few weeks I was here turned out to be critical constants and beacons throughout the next 6 decades. The first of these was Ralph Yount, who was assigned my academic advisor. And the second was Susan Anderson, who is now my wife at 58 years. I don't know when I started working in Ralph's lab, but it was really, really soon after arriving, and then throughout my undergraduate career and several summers. And he gave me a desk in the lab that made me feel really special and part of a really fun research group. You saw Dick Barclay and Boyd Haley earlier. There's also John Herman and Donner Babcock and Kay McCullough and Bernie Jones and probably a few others. But it was really a fun group and a lot of camaraderie, as was pointed out earlier. And I tried throughout my own career to encourage that sort of camaraderie in my own group. When it came time to start learning how to do research, I didn't realize how rare it was for the major professor to actually train an undergraduate himself. Yet that's exactly what Ralph did. Rather than farm me out to a graduate student, he personally taught me how to accurately and rapidly perform pipettes with glass pipettes, rather pipetting with glass pipettes, to do timed assays, taught me how to do myosin and TPase assays, taught me how to design experiments, and of course, taught me how to purify myosin. And most of you probably know that myosin is purified from the loin of rabbits. And so Ralph would stand over me with a stopwatch, timing me as I sacrificed, exsanguinated, skinned, and excised twitching loin muscles from the rabbit. And when the second loin splashed into the icy chelator solution, Ralph would call it the time from his stopwatch. And this really was great for my research career, as it turned out, because the enzyme that I studied predominantly for 40 years was rabbit skeletal muscle phosphorylase kinase. And by the time I retired, and I did retire in 2018, I tried to correct that on the program but failed. So by the time I retired, I had splashed over 4,000 rabbit loin muscles in that same icy chelator solution. So that was a huge benefit to have had that training. Ralph had a huge influence on my life outside the lab as well. I remember one summer, well, it was a fall, that Susan we were still dating, but she had taken a job at the Western Fish Nutrition Lab in Cook, Washington. And sometimes after work, I was working in this lab, I'd go down at 5 o'clock, jump into my car, drive the 300 miles on 2-lane roads to Cook, see Susan for an hour or so, turn around, drive the 300 miles back, pull in the parking lot at 8 a.m., go up to work. One time I performed this feat several times in the same week. And Ralph called me into his office and said, Jerry just married the damn girl, which I did after our junior year. And that was very good advice. After Susan and I graduated from WSU and the short stint in the Peace Corps, ended up back in Pullman to earn some money working as lab techs. And I was working for a pharmacologist, and it took me not long to reach my own conclusion that probably the stuff that I was doing was pretty sketchy, and I didn't want to do it anymore. So I complained to Ralph about this, and about 2 weeks later, I received a letter from Dave Metzler at Iowa State University. And Dave said, might you be interested in coming to Iowa State to earn your PhD in biochemistry? So unbeknownst to me, Ralph had, on his own, contacted Dave, his own old major professor at Iowa State, and got me admitted into the graduate program. Unfortunately, years later, I realized that due to Ralph's unflinching, disarming honesty and my own wildly fluctuating undergraduate grades, Ralph would guarantee to the folks at Iowa State that I'd either be their very best or their very worst graduate student, but he had no idea which. It turned out to be also a big influence on our lives, because Susan decided as long as she's there simultaneously, she would earn her PhD in nutrition from Ames. The last time that we visited Ralph was in last fall. And he reminded us then about his encouragement for us to get married all those years back. And he seemed to take some pride in that. He also, during that visit, showed an example that's always made a big impression on me. That was his love of science, not just of myosin, nor muscle contraction, nor enzymology, nor even biochemistry, but just science. And I became aware of this a lot when I was an assistant professor as the only biochemist in the chemistry department assigned to teach every course in biochemistry there was. And this induced me, going to FOSSA meetings, to sit in all these lectures where I knew nothing. And often I would run in Ralph at these same lectures. It was late afternoon, really sparsely attended, but yet there would Ralph, he'd be there. I was there because I felt I needed to be there. Ralph was there simply because he wanted to be there. Ralph Yount has held my unwavering gratitude, admiration, and respect ever since I was a teenager. So real quickly, let me tell you one simple story. It should be quick and very simple. It's about another large enzyme. So Ralph always pointed out to me that myosin was big. So we studied a big enzyme too. And I think the enzyme I mentioned, phosphorylase kinase, is down there in the bottom of the glycogenolysis cascade activation as the part of the flight or fight response. It's the first protein kinase ever discovered. It's the only kinase that will phosphorylate glycogen phosphorylase to give phosphorylated glucose to then go through energy production to give you more energy to sustain muscle contraction, both triggered by the same calcium signal. I said the enzyme's large. It has 16 subunits. The alpha, beta, and delta are regulatory. The alpha and beta are homologous. Different inserts, these are all multiple phosphorylation site inserts. The delta is calmodulin. It's an intrinsic subunit always, even in the presence of chelator and quite a bit of detergent, it's bound. This whole complex sticks tightly together, especially in the non-activated form. And the gamma subunit is just a typical protein kinase, except that the C-terminus has a calmodulin binding site. So if you put these things together, you see it's basically a regulatory machine. It's 90% regulatory. So the enzyme, as you isolate it, is under huge quaternary constraint, as Vmax is. And almost anything you do to perturb structure activates it. And so that's been most of our career. We're studying, my career was studying its regulation. But we became more and more interested in what structure we could claw out of this 1.3 million Dalton complex. The molecule looks like this in almost every small angle X-ray scattering. It's the best model. Atomic force microscopy, scanning tunneling, transmission EM, scanning transmission, you name it. It's always a bilobal bridge structure. Bridge structure. It usually was bi-bridged, and until 2005, we did this first low resolution blob structure of it, 17 nanometers by 210 nanometers. But we saw 4 bridges, and we'd already published 4 bridges as what was essential for what we saw in terms of packing and some of the protein chemistry work we did. The position of the subunits are just estimated from immunoelectron microscopy using monoclonal antibodies. So we know the epitopes, but it's rather rough. But what I'm interested in for the rest of the talk is the beta subunit. This is a key regulatory subunit. This is the one that's coupled the most with activity. In fact, we can tell the activity of the enzyme very closely just from different conformational assays on the beta subunit, even though gamma is a catalytic subunit. So the beta is quite a bit known about its covalent regulation and its structure, its primary structure. But nobody knew anything about its secondary, tertiary, quaternary structure. So around 2010, we decided it might be kind of fun to see what we could, again, claw out of this for getting more higher order structure for this key regulatory subunit. So the very first thing we thought, well, how about we just start out, go work our way up from tertiary to quaternary, see what we can find. So secondary was pretty easy because we were able to form small amounts of an alpha gamma delta trimer from insect cells, and wasn't enough to do much protein chemistry, but you do activity assays. And this calcium dependent was identical to the holoenzyme, making us think that was a valid model to be looking at the alpha gamma delta structure. And when we subtracted beta from that, we found out that beta was about 80% helical, about 12% beta sheet, and then some random and turn. But it was an incredibly helical protein. So they thought, well, let's go ahead and try to predict something for the tertiary structure, again, in the context of the alpha, beta, gamma, delta 4 hexadecamer. So this got to be a little bit challenging in the end. We initially worked, this is of course before AlphaFold and all the fancy stuff, but we used iTaser with Yang Zhang at University of Kansas, and he had won the previous 2 CAS competitions, and his threading program was pretty good. And so we were able to have to do some ab initio calculations for the N-terminal 40 residues, but there was what looked to be a GH15 family glycohydrolase multi-domain complex, not complex, but structure. And then after that, in the last 40% of the beta subunit, was what looked to be the alpha subunit of protein phosphatase 2A. Well, to kind of test to see whether we believe the glycohydrolase structure, we took a transition state analog inhibitor of alpha glucosidase as a carbose and see if it interacted at all with the PHK. It bound incredibly tightly. It activated the devil out of the enzyme, and it caused large conformational changes in the beta subunit, based mostly on different chemical cross-linking patterns you saw, plus or minus a carbose. So that gave us some belief in that part. But we wanted to kind of narrow down the docking. Obviously, you have different ways to fold and pack these things at the end, and we wanted some physical constraints. So we thought maybe we could use intra-subunit chemical cross-linking to tell us something how it might be, those 2 domains might be packing better. And we screened a number of cross-linkers, but you want something that's short and rigid rather than a long, floppy one. So what we used that worked out really highly selective for the beta subunit and short is difluorodinitrobenzene. And this is anywhere from 3 to 5 angstroms normally on this cross-link, and of course it's rigid. And so when we did that, we found all these different beta conformers, or complexes rather. Don't pay much attention to the beta 4, which would have been a dream come true to find that right away. But if you look at the identification to the monoclonal antibodies, that apparently when the alpha cross-links, we're hiding the epitope. Because the free alpha is really aminoreactive, but the bound alpha is not. So that beta 4, I wouldn't trust that at all, and we didn't. But we did go ahead then and take the monomeric beta and do an in-gel digest on that, and found 2 peptides that seemed to be cross-linked from the end of the C-terminali. Then we did MSMS on that and found out there was a C-terminal lysine that was cross-linked either to this one N-terminal tyrosine or an N-terminal lysine. And this then gave us a way of bringing the complex together to see how it might fold. So that gave us then a secondary structure that was pretty good. We thought a tertiary structure that whoever knows really how to get the crystal structure, but one that we were proud of being able to at least see that glucoamylase probably activity. And then we needed the quaternary structure. So as I said, that beta 4 we saw with DFDNB, we couldn't really trust it because of the diminished immunoreactivity. So we went over to Carol Robinson's lab in Oxford and played around with her jazzed up nanospray electron mass spec. did some top-down mass spec, but using the phosphorylated enzyme that's really activated, so the quantity constraint is released. When we did these experiments with the non-activated enzyme, we got only the 1.3 million Daltons. But with the phosphorylated one, under non-denature conditions, we were able to see these beta-4 tetramers, the different masses depend on the amount of water in the solvation sphere on this. So we had some direct proof that there's a beta-4 tetramer at the core. So we learned then in this work the structures and the whole complex was this beta-4 tetramer that encompassed the interior of the lobes and the 4 bridges, and that the alpha gamma delta trimers were then arrayed around that. It's kind of interesting because when everything we saw in the microscope, the dihedral angle between the lobes changed a lot when you activated it, either small angle X-ray scattering or different types of electron microscopy. And most times when you have a tetrahedral protein, so this is the tetrahedral with alpha, beta, gamma, delta protomers, you get a large rotation and a slight translation. And apparently this large rotation is what we were seeing in these dihedral angle between the two of them. So that's what I want to say about that. Thank you.
Okay, our next speaker is someone I remember doing some hunting and fishing with. Bryan Lawlis, who's currently owner of Bryan Lawlis Consulting, and the title of his talk is Adventures in Biotechnology Industry. Thank you,
Mike. Been a long time since I've been here, but the campus has changed. So I'm hoping that sometime they'll rebuild Fulmer Hall. It's looking pretty ragged compared to So anyway, let me just start off by saying this is the wrong title. The correct title is that Ralph Yount had accused me of wildcatting in science when I defended my thesis. And that's pretty accurate, you know. It just kind of suited my ADHD personality and my broad interest in science as I launched my career a few years later. My background is that I started off in a group called Biocatalysis at Genentech. I worked with Jim Wells, a number of other really great scientists, Dave Estelle. And later, when I came back to Genentech, I went into process sciences for biopharmaceuticals, and after that, started up a contract biomanufacturing company, did a drug delivery company for a while, and then did some work in biosimilars. Currently, I just do some consulting and advising companies, and also sit on a number of board of director roles in the last 14 years. So I got to back up and say something. This is not, process sciences is a conglomeration of skill sets that make up these groups. It's all based on fundamental foundations: protein chemistry, biochemistry, cell physiology. That's what really makes up any good process development, process science organization, which Mickey you came out of Chiron, so you're very familiar with, Mickey, but I actually ended up running that organization at Genentech. And our job was really to take what was generated in research and convert it into a product. So I don't have any basic research to talk to you guys about. I'm talking about how to get a product out of some very early science at places like Genentech. So basically, we had 3 customers, which was we supported and collaborated closely with research and development, particular clinical groups. We transferred production processes into manufacturing, and we also had a customer was the FDA, which was really the important one. You had to file a really good CMC section, or you were going to get in trouble. In fact, you could get the whole company in trouble down the road. So it was really important. We were very rigorous in our science as we presented to the FDA our results. So my whole career is about the early part of biotechnology, that 1980 to 2000 kind of time period. And at the very beginning, the only thing we had was intracellular production of proteins in E. coli. And what a mess that was. The intracellular environment in E. coli is reducing. You can overload the chaperonins in the cell, and you get these wonderful things that look like little stainless steel ball bearings. They're called inclusion particles. The biochemical problem you had, or the problem you had to solve, was to convert a hard-boiled egg, ovalbumin, into ovalbumin from this denatured mess. And that's the only way you could make proteins back in the early 80s, late 70s. And I actually remember being in molecular genetics class with Mickey and Jim, 1978, spring, was that right? And we were having a discussion about this little known company called Genentech, which was trying to make insulin. They make the A chain in one cell line, the B chain in another cell line. And they even had they made the B chain, I think, as a fusion protein. They did a cyanogen bromide cleavage. What the hell is that? You know, what a mess. And it took, you know, explosion-proof rooms and all kinds of stuff to do those kinds of production processes. And I just said that, you know, we all agreed they'll never do this. It'll never work. And little did I know, I got hired at Genentech just 3 years later, 2.5 years later. And that's the way we were making stuff at that time. Later on, the technology evolved, and we started doing all kinds of things like getting secretion in E. coli. And actually, I tried to give credit to some individuals who did the work in CHO, 1981 to 83. They were starting to express proteins in CHO. And I think I mentioned there, yeah, Diane Panika, Art Levinson expressed TPA and CHO, and Bill Bennett and Gordon Vihar purified it, and I actually saw the first gels of purified TPA, first protein that ended up being a pharmaceutical out of CHO, which was a pretty cool moment. So the 90s, things progressed. Everybody could make stuff in monoclonal antibodies and hybridomas. We actually, Dennis Slayman came to us and convinced us to partner with him on Herceptin, HER2, for treatment of HER2 positive breast cancer. And we went on to make materials. We bootlegged that project at Genentech. Management was really against that project in research, but there was a few champions in the company that worked hard to get us to go ahead and move that project forward. We got a signal in 30 days from patients who were unfortunately very sick with HER2 positive, and which was the trigger for us to progress forward. And I would go in about once every 4 weeks or so and argue with Bill. We needed to do another run. Bill Young, who was essentially the COO of Genentech, and I'd eventually convince him to do a run, and we'd go do a run in the production facility and use it to supply these clinical trials. This all led to Lin Presta at Genentech humanizing the first monoclonal antibody, and then we were off to the races there once we got the Vacaville facility built a number of years later. But all that kind of happened all at once. So currently, antibody production, you don't need a fleet of PhDs like we had back then. You can do it. It's standardized technology. There's a ton of places you can contract manufacture at to get it done, and it's pretty routine stuff. And that was the objective, is to make it routine, as much of this as you could. So backing up to my career, first thing I had to do was in biocatalysis, we were focused on industrial enzymes to make active renin. So I made grams of active renin using really kludgy approach of the refractile bodies, as I mentioned earlier, and we could manufacture enough to make several hundred pounds of cheese, and yeah, it works, but it was pretty obvious. I was beating my head against the wall. I worked on that for a couple of years and really couldn't get this to be economically feasible. We were making it probably at scale in the order of hundreds of dollars per gram. Our target was single dollar per gram kind of numbers. So tough row to hoe. But I went to the head of research at Genentech, and I convinced him we needed an expression system in Aspergillus for secretion. Aspergillus makes proteins at very high yields, secretes tens of grams per liter of product. So we went off and developed an expression system in Aspergillus, and we started making the material there. And it was folded properly, and it was active, and so everything was looking pretty good. Subsequently, I found out that they'd sold that technology to Chris Hansen's here a little while back, which is a renin production company, and they had all built a system in trichoderma, another fungi that really secretes proteins. It'll make cellulase at 100 grams per liter, which is pretty amazing. So, see, I can hardly see these slides. But at Genentech, as we moved into the humanization, we were really doing large scale, and we went on to build quite a capability there, but I'd say subsequent. Hold on just a second. I can't read this. Going to stepping down's a problem at my age, but. So this was the patents, one of the patents we did on Aspergillus. There was a variety of those, and that kicked off the whole production capability there. I then moved on to subtle lysin production. Very straightforward. It's a big market. It's $3.5 billion in sales, projected to be this year. And we did that in B. subtilis. We had incredibly productive fermentation process, half a gram per liter hour secreted product, and we had to manufacture a 4% solution that had the appropriate yield, suitable purity for its use. And the biggest problem we had, honestly, was removal of solids. When you have these high cell mass levels and the kinds of substrates they put in the fermentation, it was 60% volume solids. But just finding the conditions to purify it so it didn't auto-catalyze, tear itself up, was kind of tough. But we got there, and we made up material, and we put it in a 35,000-pound tanker car, shipped it off to Procter & Gamble, and they made up some Tide, liquid Tide, and they got to the bottom of the bottle, and this big brown glob fell out at the bottom, and it was pectin. So my job, and I had to do this very quickly because that was the only income we had at Genencor, was this relationship with Procter & Gamble, was to find a way to separate the subtle lysine for cents per gram from all this pectin. Well, the engineers came to me about a year before and said, hey, we got a problem. The enzyme came out of solution while we were concentrating. So I went in and I looked at all their materials and stuff. Immediately, because I'd crystallized ribulose bisphosphate carboxylase in my PhD work, I immediately said, well, you made crystals. And so I said, well, we can take advantage of that now. And I figured out the conditions for crystallization, and we were doing that at 500 pounds of crystals every 24 hours to satisfy this situation. So that's actually a slide out of the patent for the crystallization of subtle lysine. And just for fun on the side, early on, Dave Estelle and where's Jim? There he is over there. Dave Estelle and Jim had made, this was with Dave, wasn't it, with Jim? Yeah, it was the M222, which was sensitive to peroxide oxidation, inactivated the enzyme. They converted it to an alanine, and I purified up about a quarter of a pound of this and had it sitting on the shelf, big columns, and it was the old-fashioned purification. And the guy from research, the crystallographer, came over, and he said he wanted some enzyme. He heard I had this, wanted some enzyme to make crystals. And I said, well, how much you want? And, he's used to getting like a milligram of stuff. And I pulled out this jug off the shelf of lyophilized material, and I started spooning out into a 50 milliliter screw cap, 50 ml screw cap. He said, okay, that'll do. And he just about fainted. He was really impressed with that. But here's the differences I came back to Genentech in '88. Here's the differences in shipment containers between Genentech and folks at Genencor. And this was one of the first products I worked on when I got back at Genentech, was Pulmozyme for treatment of cystic fibrosis. It's recombinant human DNase. And the problem we have there is very short timelines. And we got to phase 3, and we needed to scale this because it would never work at the scale we were operating at, especially considering the indications we were going to go into beyond CF, we thought. So we had to go from the 1,300 liter reactors to very large scale, 12,000 liter reactors very quickly. So we had a nice process that Bob Breidenbau did the purification on and figured that out. And he was in my lab at that time at Genentech. And he sorted that out, and we were operating that at 1,300 liters, a few hundred grams per run. We needed to go to a few kilos per run. And at that time, the problem was we needed to build a new plant. We needed to scale to that plant. So all of those are changes for the FDA, and you can't switch back then in the middle of phase 3. So I just said, screw that. Their idea was process defines the product, so we're just going to show that the process is the same at the two scales. We're going to prospectively tell them what the protocol is going to be for that, and then we're going to do it. Well, this was 1992, and they didn't allow that back then. So we went in, we did it, we got it approved, and we did the whole thing from idea of the product to finish in about 3.5 to 4 years, which was record-breaking time back then. I think it is still today for products. But this is the way they do it today, is you file a protocol, prospective protocol with the agency, rigorous, which is what I learned from Ralph Yount and Bruce McFadden, rigor in your science, and then you go do it. And that's how we did it. So a little homage to the Palouse here with the picture on the right. But what I got out of WSU, first off, the students and the faculty, just a huge, it made you, people were really good, and it just made you perform better. Made you more rigorous in your science, because you wanted to be part of that group, that, you know, how those people thought. Very strong foundation in protein chemistry, enzymology, and that was very useful to me throughout my career. And really, Mike, I think you captured it a little earlier. Ralph was key to the culture and getting all this started. And so was Bruce. He was very powerful influence. As a graduate student, you couldn't find anybody more supportive than Ralph. Very empathetic, a good guy, and he really, you know, enforced critical thinking. Now, I wasn't a faculty member, and I'm sure the relationship might have been a bit different. You might get bludgeoned with brute logic if you were a faculty member, but the guy was super, super supportive for students. And I just found it a great environment to enable me to do what I've been able to do in my career. So thank you.
So our next speaker is another former student, Mickey Urdea, who was the founder and partner for Halteres Associates, and his title of his talk is A Random Walk Through Biotech Shiny Objects.
Well, I'm very glad to be here to help celebrate the memory of Ralph. I mean, Ralph was key in my life, as so many people here. I was at Northern Arizona University. When I was graduating, I had choice of a few different grad schools, but I came here mostly because of Ralph. Ralph called me, and he just convinced me this is the place to be. In particular, I really liked the approach of having a very strong chemistry background to do biochemistry. So, and that's what it was. When I got here, I found, you know, just an incredible group of people. And I was really impressed by the collaboration that occurred across different groups. You know, physical chemists working with inorganic chemists, working with biochemists. And I actually ended up working for Ivan Legg, who was an inorganic chemist who loved metallobiochemistry. So, you know, here, I got a degree in biochemistry with an inorganic chemist. I don't know where else I could have done that. So I just found it to be a great place to work. You know, one of the things that I remember as an experience here that was very beneficial is the seminars we had to give. And many of the faculty showed up. Someone was talking about that earlier. But those things were rigorous. And we just got grilled by everybody very intensely. We were all freaked out to get up the first time to do them, I got to say. Jim and I were talking about that yesterday. I mean, it was really something. But did it ever prepare us, you know, to leave? And we are grilled like that every day of our careers, still to this day. And that set the stage for us. I think Ralph created a culture of tough love here. It was, you know, they love you as long as you're producing. And, you know, do what you say you're going to do. And I appreciated that. And we all loved working very hard. You know, the lights were on all the time because we were in there. And that was great for our careers after we left here as well. Ralph also helped me get a postdoc. I wanted to go to San Francisco to work for Bill Rutter, who at UC San Francisco had created this great biochemistry department, and also set the stage for biotech companies. And I sent my note in, and Bill didn't know who Ivan Legg was, but he knew who Ralph was. So fortunately, Bill called Ralph and asked him, should I take this guy? And somehow he convinced Bill to take me, so I'm ever grateful for that. I also got a chance to come back here a number of times after I graduated because Ralph had an NIH biotech training program. So I got to come back and I met some of the new people here, and it was just a great interaction with people, talking about what's going on in biotechnology, keeping up with WSU. But after I left academia, I went to Chiron Corporation, a biotech company that was actually started by Bill Rutter. And I was one of the very first employees there. And in that company at that time, this was during the HIV/AIDS epidemic, and that was the place where HIV was first cultured, cloned, and sequenced. And also, we had developed the first HBV vaccine, and we also had the discovery of both the hepatitis delta virus and hepatitis C. That won our colleague Mike Houghton the Nobel Prize. So, in all this situation where all these viruses were swimming around, I had this idea that it would be useful to quantitate viruses in blood. And I thought this could be used for therapy, and you'd be amazed that I would go talk to virologists and physicians, and they would say, that is a really stupid idea, Mickey. But fortunately, Bill believed in it, and he gave me the resources to go ahead and work in it. And if I go to figure out how to use this. So I was able to put together a group to develop technology we called BDNA signal amplification. And I'm not going to go through all this stuff in here, but I just want to tell you this was really hard. It took us almost 8 years to do this. And one of the people that was key in this was Brian Warner, who was also here in Ivan Legg's group as well. So he was a key contributor to all this. There are a number of inventions here. One had to do with the way in which we use probes. We invented these branched DNA molecules. We had, in fact, non-natural nucleotides that were in some of the probe sequences. And we had this new chemiluminescent substrate for enzymes. And at the end of the day, we were actually able to get 24,000 labels onto each genome of HIV RNA. That gave us the signal that we needed to detect it at very low levels, down to 50 copies per mil of blood. That ended up being important. So when we were ready with the assay, we contacted a guy named John Mellors, who I knew at the University of Pittsburgh, and he worked in the AIDS clinical trial group to collect samples from people that were infected with HIV. And he did this over a 7-year period. And he said, okay, I'm going to send you guys some samples. So he did. He sent us samples from 10 patients, and we didn't know what he sent us or what they were. But when we got the results and we sent them back, John called very excitedly, saying that you guys won't believe with this. So what he sent us were samples from 5 patients on the top there that did not progress to AIDS from HIV infection over this period of time. And all of those kept their CD4 counts high. Now, the definition of AIDS at the time was if your CD4 counts go below 250 copies per mil, and I have a red line in there, that means you have AIDS and all the complications that come with it afterward. So on the top, all those people kept above that line. And what was surprising was the RNA from the HIV and the black dots there, they all stayed low. But in the people that progressed to AIDS over that period of time in the lower set, you see their CD4 counts drop to a point where they go below the red line. The RNA came up in that same time frame and came up very high. That was a big surprise at the time. You look back and go, well, that seems so obvious. Well, it wasn't obvious then. So at that point, we knew that there was a correlation between RNA levels and progression to AIDS. There were a lot more studies beyond this that were done, and it really got nailed. And But the thing that we didn't know yet was, if you artificially lower the RNA level, does that decrease the likelihood of developing AIDS? All right? So that was the next group of things that occurred, and one was a set of studies we did with David Ho, who was at Rockefeller at the time, and David and his colleague Alan Perlson figured out the kinetics of RNA drop using our BDNA assay, which we had supplied to David. And he found that no matter what your starting level of RNA was, with the very first drug that was available, ritonavir, you got these rapid drops in the RNA levels. So this was carried on beyond this for years. To this day, these studies are still being done. But what we found is that if you got to a point where you drop the RNA below 50 copies per mil, that person is not likely to develop AIDS. And it took modifications of the drug therapies from monotherapy, which this was, to having 3 drugs. And what was referred to is highly active antiretroviral therapy, HART as it was called. And with HART then we were able to get there. And on the right-hand side was a study that was published in, I think, 2018, where it looked at the death rates from AIDS in the population at different age groups until HART was developed and introduced in '95. And look at the drop in all those. That was all done by conducting both the clinical trials and the monitoring with viral load testing. So that was quite something. And so what ended up happening was that HIV viral load became a clinical surrogate endpoint. And I'll explain what I mean by that. It went from being a point where people had a death sentence to it being a manageable chronic disease because of that. So here I'm just showing not using a surrogate endpoint and how you normally do a clinical study. You have a disease, let's say it's HIV infection. You have some sort of intervention, antiviral drugs is the example here, and over time, you look for the clinical HART endpoint. That would be AIDS. That takes a long time. It's a lot of money and a lot of patients. But once we had convinced the community that HIV viral load was a surrogate of that is, if the RNA goes down, you're not going to get AIDS. So that's the endpoint for the clinical trial. That's the clinical surrogate endpoint. And now you get much shorter trials, many fewer patients, and much lower cost. So this has been the theme for my last 40 years, is clinical surrogate endpoints. And they come in all forms. But I must say that the best surrogate endpoints with the best drugs mean that at the end of the clinical trial, you don't need those markers anymore. They can become a diagnostic maybe or a surveillance marker, but you don't need it to manage. But if your drugs aren't that great, you need them to manage as well. So it took a long time to get to a point where people didn't really monitor with viral load for some of these diseases. But here comes my random walk. I've had a chance to work in many different disease areas since then. When I was still at Chiron and then Bayer up at the top there, we went on from HIV to hepatitis C and hepatitis B. Those became clinical surrogate endpoints as well. I had a foundation called Catalyst Foundation for Health that was funded by the Gates Foundation. We did many biomarkers, and we were trying to find a surrogate from MTB, for MTB. And we didn't find one, and one still doesn't exist today. You'd think it would be, because if you find the DNA in sputum and that disappears with therapy, you must be cured. Well, what we found over 15 years of studies was that a lot of those people still have nodules of MTB in their lungs. They're not cured. So we're still searching. I started a consulting group and represent that here today, Halteres Associates. We did projects with many startup companies. We've done studies with about 250 companies to date, startups, large diagnostic companies, NGOs as well, Gates Foundation, FIND, WHO. And when I prepared this talk, I really hadn't thought about the fact that, well, a lot of these things are surrogate endpoints. And so I went through and I realized so many of these things were on the way to being, or we helped get it over the goal line. Some of them involve inventions from me. Some of them I'm just a consultant with my group for that. But most of those you see are pathogens, and most of those really are used in the trials. Some of them are still used in monitoring, like for some of the NTDs. But some have not been approached yet, and all the ones in red there, which are situations of infectious disease, they don't have a clinical surrogate, and because they're much more complicated. For instance, most of those would require panels of tests. So those are still going on. But it's not just infectious disease. Clinical surrogates can be used in a lot of other disease states too, like non-communicable diseases. And I started a company called Halteres Biosciences that actually developed a clinical surrogate for type 2 diabetes. And this would be looking at people that don't have it and have a high risk for getting it. And we found that we could create a score using 7 markers, and that when we looked at the what happened to the markers if you, for instance, had lifestyle changes or treat people with metformin, those markers went down. And we found trials where we could prove that that was actually decreasing the risk to go on to type 2 diabetes. We had 2 other things in blue here that I didn't get a chance to finish, but they were on the path as well. And then Halteres and non-communicable diseases, we're doing all kinds of things that haven't been completed yet. And that's because a lot of those diseases, like Alzheimer's, the biology isn't very well worked out. I mean, people today are talking about p-tau 217, all the other things, but when you really look at it, that's only defining a portion of the disease state. So I think we're still looking for the right biology there. But what I do want to talk about now is cancer. I'm doing some work now, so this is my second thing that I want to talk about more deeply. And it has to do with, strangely enough, hyperpolarized metabolites for magnetic resonance imaging. So I'm doing a lot of work in this area for the last couple of years. Let me explain what it is. So standard MRI imaging tells you about morphology, but when you use these hyperpolarized metabolites, you can actually figure out what is metabolically occurring within that mass. So the way that's done is to look at metabolites, and I'm showing a few examples up there, that you know how they are converted to another product within the metabolic state, and that's different in the disease state than it is in the healthy state. So a good example of that, and on the table on the right there, is pyruvate, which is used as the probe. And in cancer, more of the pyruvate goes to a lactate formation than it does to go into the TCA cycle. So you can actually look at, if you do this labeling properly, a ratio of lactate to pyruvate as an indication of the cancer state within a mass. So I'll show you an example in a moment. But the way this is done is people synthesize pyruvate with an enriched carbon for carbon-13, and then we use hyperpolarized hydrogen, where it's taken to a very low temperature with a catalyst, and you get an alignment to all the spin states. And that can be transferred to another molecule in the appropriate conditions. And so you can transfer that to the pyruvate. That now lights up the pyruvate in a magnetic resonance image. So you get about 100,000-fold increase in the signal to noise as a result of using that approach. I mean, it's really incredible how well it works. So I'm going to give you an example here. And I've mocked this up a bit, but it's based on a study that was presented at UC San Francisco earlier this year by Stephen Lai from MD Anderson. And the top there is a dilemma that physicians often run into, and this is using standardized MRI. So this is an example of a lung cancer, where you now see a nodule with that arrow, and then it's treated with an appropriate chemotherapeutic agent, and it's larger at 1 month. Now the dilemma is there's more than one reason that could occur. One is because the cancer is getting worse and it's getting larger because of that, or it's been infiltrated by immune cells and it's bigger because of that. So you can't tell which one it is based on the morphology. That's why people wait much longer to see what's going to happen. Is it going to get better or is it going to get worse? But what Stephen showed was that if you now use the MRI imaging with hyperpolarization in the bottom left, and I hope that those white dots and things show up a bit, but that is the area where the cancerous activity has been noted because of the high lactate to pyruvate ratio. And then they treat it, and the second one, you don't see that anymore. That therapy is mostly working. And so when they went on and looked at this particular situation, the tumor disappeared after some period of time. They knew what they were doing as a result of that imaging. And this is now being used in a number of studies. A lot of them are still preclinical in mouse models, but what Stephen showed were in humans. This is going to be used in many different cancer states: prostate cancer, pancreatic cancer, solid tumor cancers. It really does appear to be a general marker. I think this is on the path to becoming a clinical surrogate endpoint. So I think that, like I say, this is on the path, and I'm actually working with 4 people at UC San Francisco, 2 companies, and a group at Salk. And I'm trying to coordinate the clinical studies for all of this. I've got a good grant from a company to do this, and I'm really quite excited about it. I think it's going to be a major advantage, a step forward. Lastly, all these stories and more can be found on the Halteres YouTube channel. We have done a whole lot of audio podcasts, 32 to date from our first 3 seasons. We've got a 4th season coming out in a couple of weeks. We've had a video podcast. There are 32 episodes of that also there, and a new podcast that we call This Is Only a Test. There are lots of different subject matters there. I'm going to provide people here links to all of them and what they are, so you can get them if you want. I thought there might be some students here and they might enjoy it, but anyhow, that's what keeps me off the street. So thanks. And thank you, Ralph.
Okay, so we'll take a 15-minute break and be back at 3 o'clock.
All right, well, my name's Jim Wells. I'll be speaking at the end of this symposium. I'm so glad everyone was able to come to this very special event. Ralph from up above will be looking down at us very gratefully and seeing all of his friends here. And it's a pleasure to introduce Lin Randall, who's now at University of Missouri. She was on the faculty here, as you heard from Ross, from I think it was 82 to, 82.
81.
Right, I think I was leaving just at the time you guys arrived. You guys were the big fish that were recruited to WSU. So it's a pleasure to have you both here, and Lynn's going to give her talk.
Okay.
Advance your slides here,
Okay, yes, I think I see it. We'll try this. Yeah.
There you go. Okay.
Okay, thank you. So I am happy to be here to honor Ralph, but very, very sad that he won't be with us. And I hope I don't break down into tears, but I may. Now, Ralph really did support me from the beginning of my career, and he really had faith in me and kept me going. So, let's see. Oh, I see, I've got a copy. So, yeah, I first met Ralph when I was interviewed here. And then 5 months later, I accepted the position and then arrived, and Ralph was there to welcome me. And he started out by offering me advice that I didn't think was very appropriate. So I did a lot of my research work in vivo. And I did a lot of it by running SDS gels, and I learned a whole lot that way, and actually became prominent in my field. But Ralph's first advice was that I couldn't learn, how did he put it? I couldn't learn very much by running SDS gels. And what was the other thing? Oh, you can't just look at bands on gels. He actually told me that right out. And so, as I'll explain, I took his advice, but only in part. I continued to do what I thought I did very well, work to resolve the process of protein export in vivo. And I'm going the wrong way. I'm nervous, everybody. More nervous than any talk I have ever given, and I've given a lot of talks. But the thought that it's for Ralph is really distressing because he's not here with us. But in any case, he did mean a lot to me. And so in the early years, as I said, that I did a lot of my work in vivo, and I did it by running SDS gels and analyzing them. And Ralph advised me that that was not a way to do research. He said that I needed to purify proteins and to work in vitro. So, what's this one say? Oh. Somehow these slides aren't what I thought they were, Jerry. Anyway, I did take Ralph's advice in part, and I didn't do all my work with SDS gels anymore. I actually became fairly biophysical. So the system that I studied was protein secretion, and I studied it in E. coli, the gram-negative bacteria. It's not my blood sugar dropping or anything. I've had diabetes for 53 years, something like that, and so little alarms go off when my blood sugar goes up and down. So this is essentially the cycle that was determined for the cycle of protein export from in the gram-negative bacteria. I think I should give up, Jerry. Maybe this is it. Well, the protein is synthesized in a precursor form with a, oops, I thought that was a pointer. Is one of these things on here a pointer? No? I can't point. Okay. Well, you can see on there an illustration of a polypeptide being synthesized with an extension on its amino terminus, and that's the leader peptide. And what we showed over the years was that the leader peptide, the presence of it, retards the folding of the polypeptide. And the folding can only go through the translocation apparatus, which is shown up in the top there in the membrane. It's the SEC-YEG complex that forms a channel in the cytoplasmic membrane in E. coli. And the protein is brought to that channel and extruded through it, but it has to remain unfolded. And so one of the very first important contributions I made was showing that there's a small chaperone in the cell, the product of the SEC-B gene. It's a tetrameric protein. And when this doesn't point. So it's shown in green there, and it's labeled SEC-B. And what it does is it binds very tightly to unfolded proteins, but it can only bind them when they're unfolded. And then SEC-B itself interacts with the protein SEC-A, which is another secretory protein. SEC-A functions as a dimer, and it's an ATPase. What it does is it binds the SEC-B carrying the precursor delivers it to the translocon, and uses the energy of ATP hydrolysis to pass the protein through the translocon. The translocon is shown up there at the top in the membrane. And there's a protein that extrudes through the membrane with its active site on the outside of the cell, and that is the enzyme that removes the leader sequence from the secreted protein, leader peptidase. And so I did a lot of work determining that cycle, and I did it in vivo on SDS gels. So good thing I didn't know Ralph when I started out, or I might have never done anything. But I did very successfully work in vivo and did a lot of work using SDS gels. But I did move on to taking a little bit of Ralph's advice. Let's see, next slide. This one say. This is, I'm sorry, but I can't read it from here because it's at an extreme angle. Maybe, Jerry, you should come up here and pass my slides to me. Maybe I should just go down there. Could I just go down there? Yeah. I saw someone do this. Yeah, somebody's got to help me go down the stairs.
Yeah.
Yeah.
That might be easier. Okay, let me take that.
Because I can't
Boop, boop. Here's this. Okay. You want to come this way, down the stairs? Okay. Okay. Step. Boop, boop. The maestro here. Okay, there, and you've even got a pointer right there. So,
No, but if I pick up this pointer, I fall over. All right, this is the
okay.
Okay, correlation of competence with a lack of structure. Oh, okay, can you hear me now? All right, so this is some of my early work. Showed that the protein has to be unfolded to go through the translocon. And these were in vivo experiments done on SDS gels. And here you see the precursor maltose binding protein. It's being synthesized in vivo, and with time, intracellular. It's disappearing because I've added protease, and when it's unfolded, it gets degraded. And here you see the export of the mature protein, which has had its leader removed. And and it folds, so protease doesn't degrade it. And here the unfolded precursor, you see is completely degraded when I add a protease. Yeah. This is one of my favorite experiments that we ever did. I believe that Sec-B bound the protein when it was unfolded and blocked folding. Can you hear me? I should just quit, right? Everybody vote. Do you want to hear me, or shall I just sit down? Okay. All right. This is one of my very favorite experiments that we ever did. I believe Tracy Topping is in the audience, right, Tracy? Yes, she's there. Okay. Tracy was learning to do fluorescence because we followed the proteins folding by fluorescence on the advice of Ralph Yount, that I needed to get a little more biophysical and find out what I was doing. So Tracy had been doing fluorescence to watch proteins fold, and this is the folding of the maltose binding protein, which is one of the ligands we used a lot. And so Tracy was sitting at the spectrophotometer. You remember this experiment, Tracy? Yeah, this was really exciting. So she's watching maltose binding protein fold by jumping it into the cuvette and following the fluorescence. This is with no addition except the maltose binding protein. Well, while she's doing this experiment, literally, I get a shipment from, Bill Wickner. Okay, now Bill Wickner studies protein secretion in E. coli, and he had purified the Sec-B protein, which is a chaperone that binds unfolded proteins. And so we had proposed that Sec-B binds them to block the folding to keep them competent to be exported. So Tracy's sitting there doing this experiment, dripping it in, watching it fold, and so she's just about to start the next experiment, and I say, wait, Tracy, before you drip the maltose binding protein in there, first empty the cuvette and put some Sec-B in there. So Tracy puts Sec-B in, drips the unfolded protein in, and it doesn't fold at all. And we had discovered that Sec-B binds the unfolded protein to block folding, all in a matter of minutes. It was the most exciting experiment I ever saw happen right before my eyes. And
Ralph was right. Ralph was right.
Right. No, Ralph was right. I mean, that's why I said I took his advice in part. I still ran gels, but I started doing biophysical experiments. So we studied protein folding by fluorescence, as well as learned to do that from Bob Matthews, who was a protein folder. I went to Wayne Hubbell's lab and learned to do EPR and did folding by the changes in the EPR signals. And something else. What else did I do? There's a third one. What's another one, Jerry? Analytical ultracentrifugation. Yeah, I went to Ken Van Holt's lab, bought an analytical centrifuge, and had many years of fun doing analytical centrifugation to study the system. And they all led to these same conclusions. So, and here you're looking at export in vivo, and I imagine these results did come off a gel. The percent of protein that was secreted when it's wild type, when there's no leader, it doesn't secrete as much because it actually folds in the cell, and without Sec-B, it's not as efficient. I'm really nervous. I don't usually get this nervous. How do I change the cell? Oh, you do? Just call for the slide. Okay. And so this was the working model that my laboratory developed, that there was a kinetic partitioning that selected for non-native proteins to go through the Sec-YEG channel. And so there was a competition between the kinetics of folding. The protein also can aggregate, combine Sec-B, and Sec-B stabilizes it in the competent state to be exported, and Sec-B is bound by Sec-A and delivered to the Sec-YEG. So can I have the next slide? So Ralph actually was on, I think, all of my graduate students' committees because he was always very serious about helping them. And so when we went to choose a committee, we had Ralph on there. And this is one of the graduations with Ralph, me, one of my students, Jiping Liu, and one of Ralph's students. And somebody say something? No. And Ralph also did a wonderful thing kind of indirectly for me. He had a graduate student who was working with Chris Cremo. Is Chris Cremo here? No. Because unfortunately, I don't know the name of the graduate student. She's was a Native American Indian, and I don't remember what. Jennifer Only. You know her. You're still in touch with her? No. No, because I would like to thank her personally. But what Jennifer Only did for me, I had just opened my restaurant. I had gone to a culinary institute. I am a trained chef, and I had time. You know, I'm only running a research lab, so I opened a restaurant. And somebody thought that was a terrible thing for a faculty member to do, and wrote to the Moscow Pullman Daily News about the restaurant I had opened. And what this person, this woman, wrote was, if I have so much time, I shouldn't be running a restaurant, I should be reading the literature. Jennifer Only wrote to the newspaper, Dr. Randall doesn't need to read the literature, she writes it. And that was amazing support from what was one of Ralph's graduate students, and I'll always remember that. So, next slide. And so Ralph really did mean a lot to me in supporting my career, and I'm sure he is missed by everybody who is here for this. And I think that's all my slides, right? So that's all I have to say, except Ralph felt he always believed in me. And he encouraged me. I didn't think I could learn to do biophysics. I was a zoology undergraduate major. But I learned to fold proteins from Bob Matthews. He used fluorescence. He taught me over the phone. Did I go through this? Wayne Hubble, EPR, and the centrifugation, analytical centrifugation, Kin Van Holtz, and Bo Demler. And so I feel like I owe most of my success to Ralph Yount. And I thought I wouldn't make this all the way through without crying, but we will all remember him, I'm sure. That's all I have to say. Thank you.
Thank you, Lynn. Do you want me to help you? Sit there. Okay. Ooh, thank you so much, Lynn. Okay, next we have Keith Dunker. Keith, like Lynn, actually arrived when I was during my stint here, and he's gone on to do some amazing work in bioinformatics, and especially the unfolded protein, the importance of intrinsically disordered proteins, something that I think many in the field, or there was no field until Keith was involved in it, actually. Keith, you want some help? Here.
Thank you. I can probably do it, but I don't want to take the chance.
No problem. All right, so here you're going to hear about this from the man himself, Keith Dunker.
Okay. First of all, I'm going to do my talk in two parts. I'm going to give a regular scientific lecture, and then I'm going to follow that with a few comments about what Ralph meant to me in coming here. Okay? So we'll do the scientific part. As Jim talked about, I study intrinsically disordered proteins. These are proteins that don't fold. They're not folded. They're floppy. They're like spaghetti. Let's go to the next slide. Pardon? Is it? Am I? Oh, okay. The trigger for my IDP research was a seminar that occurred in the biochemistry department given by Chuck Kissinger. Kissinger had been an undergraduate at Washington State, and then he had gone to University of Washington, where he got his PhD, and he became a crystallographer there, and he determined the crystal structure of a very interesting protein, and that's what he was talking about. At Washington State, I think he got his master's degree, and I think it was with Mike Griswold, right? Yeah. And so anyway, before he went to University of Washington. So he gave a seminar, so next slide. It's about the activation of T cells, and then they can do something like destroy your heart transplant or destroy your kidney transplant. And so what happens when they're activated is you get a binding of the antigen to the surface. The surface opens a calcium channel. Calcium comes in and binds to a protein called calmodulin, and then calmodulin binds to a protein called calcineurin, and that turns on calcineurin, and then calcineurin takes phosphates off of a protein called NFAT, which stands for nuclear factor of activated T cells, and this removes these phosphates. And so then it turns out that these phosphates are in the middle of a nuclear localization signal, and that nuclear localization signal is a bunch of positive charges in an unstructured region of protein. The phosphates then interact with the positive charges and prevent it from going into the nucleus. You take the phosphates off, now it goes into the nucleus, turns on a bunch of genes, the T cells get activated, the transplant gets rejected. And it shows over there, there's a couple of drugs that bind to this protein called calcineurin, which is the one that calcineurin is a serine threonine phosphatase, and these drugs will bind to calcineurin and they'll help prevent the activation of the T cells, and so that helps to stabilize the transplant. So shall we go to the next slide? So here's a picture of calcineurin on the left, and on the right is calmodulin binding to calcineurin. Now you notice, or it's not actually calcineurin, it's another substrate, but it illustrates a principle. You'll notice that calmodulin completely wraps around its target. That's 360 degrees it goes around it. So this target has to be extremely accessible for this thing to work. Now what's not shown here, and next, click it again, is it says on here 95, but that's a mistake. It's a 96 residue segment that connects the yellow A subunit, there's a B subunit, but it connects the A subunit with this disordered segment, and then over there's a green segment, part of it, and that green segment is called the autoinhibitory domain. So this unstructured region, when it's not attached to the active site, it's active and the protein can do, but can function. But and then you see over on the left side, there's a part that looks like a helix on the red disordered region, and that's where the calmodulin binds. And so it's a completely missing region on the X-ray structure, and that missing region carries these two biological functions, the binding site for calmodulin and the binding to the active site that blocks it. And in my own research in the lab, I'd become acquainted with some disordered proteins, but here was an example that had clear function. And so after the seminar, may I have the next slide? Kissinger, hi Chuck, and Chuck is here. Chuck Kissinger and I had a discussion about this region. And we came up with 3 questions. Why don't they fold? First of all, we knew that there were other examples of missing electron density regions in proteins in the protein data bank. And so why didn't these disordered regions fold? The second question we asked was, how common are they? And the third question we asked was, what are their functions? Now, I think, and so that afternoon, I started collecting examples of disordered proteins and started to study them with bioinformatics. And I think we were the first group ever to treat unstructured proteins as a group of proteins that should be studied as a group rather than by individual things. And that's what led to our discoveries. So next. Now, we found that they don't fold because, whoops, they have too few aromatics. It turns out that aromatics stabilize the cores of proteins. They interact with each other and they really make the protein core stable. And the disordered regions that we had discovered had too few aromatics compared to structured proteins. They have a ratio of hydrophilic amino acids to hydrophobic amino acids that has way excess of hydrophilic and way deficient of hydrophobic. So of course it doesn't fold if it doesn't have enough hydrophobics. We also found that they have a low sequence complexity. We found that they have a large net charge, or they have a large total charge, but a large net charge in particular. And the last thing is that too many prolines. Prolines disrupt structure because they don't have the NH group, so they can't hydrogen bond, so they can't form an alpha helix, they can't form a beta sheet, which are the two core structural features of structured proteins. And so disordered proteins also often are rich in prolines. I want to emphasize that not every disordered protein has all of these features. It's a combination of these features that lead to disorder, and some have more of one and less of, and more of another. Okay, next. Oh, that we had this one. This is the one previously. Next slide. It's going the wrong direction. Okay, so then we developed a predictor of disorder. So it's that we don't need to worry about the details. Let's go on. Okay, so then if you predict disorder, you find that on the x-axis is the size of the proteome, and on the y-axis is the fraction of disorder. And it's not that clear, but archaea are down at the bottom and they have the least disorder, and they're the blue ones. Common bacteria have more disorder, a little bit more, then that there's a straight line across at about 30%. And then the eukaryotes are all above 30%. Okay, next slide. Okay, now can we go back to the beginning of my slides? Because there's one slide I think that got missed. Keep going back, back, back. Is there one? Where's the next one? Where's the next one? Oh, yeah, this one. Somehow we skipped this slide. And as we went through the literature looking for comments about disordered proteins, pardon? Sorry. Thank you for reminding me. We went through the literature. We found that in 1938, Karl Landsteiner, who had gotten the Nobel Prize for working on blood groups and figuring out the blood group things, and Linus Pauling, who is the outstanding protein chemist of the 20th century, proposed that they were talking about antibodies, and it's probably wrong for antibodies, but they had the idea that antibodies, they knew that there were some antibodies that could bind to differently shaped antigens. And the hypothesis they proposed was that the antibodies were unfolded and they could fold in different ways, therefore having different binding pockets and therefore bind to different antigens. And that was their proposal in 1939, 1940. The first IDPs were characterized in 1950s using optical rotatory dispersion, and then thousands have now been characterized by X-rays. The common thing is if you have a missing region of electron density in an X-ray structure, it's usually disordered. And we have this thing called the Protein Data Bank, which has all these structured proteins. And more than 90% of them have disordered regions, okay? Some of them are short, but some of them, 30% or so, are quite long. And then you have NMR as a way of determining protein structure, and many NMR proteins have regions of disorder. And then finally, the computational biology and bioinformatics. And anyway, this idea that disordered proteins can carry out function is a paradigm shift. So let's go, now we can go forward. I don't know how we missed that slide. But now keep going. So no, back one more. Okay, and so basically you can see this trend in the amount of disorder that archaea, common bacteria, and eukaryotes have much more disorder. And we have pointed out an arrow to one particular point. That's humans. This estimate is that humans have about 40%, 40% to 50% of our amino acids are in regions that are unstructured. So they're incredibly common. And that's something that wasn't realized before we did our work, okay? So shall we go on? All right. Now, we want to talk about function for one moment. I want to give one functional example. I did one with the calcineurin that we began the seminar with. That's really an important one. But here's another one. Probably every biochemistry student learns about the Lac repressor. And the Lac repressor binds to DNA and prevents other proteins from being bound. It comes off, and then the Lac operon starts going, and so on. So it was the first protein to regulate transcription in DNA. And so what happens, you see, is that the Lac repressor has a disordered region when it binds to the non-cognate part, the non-DNA that it doesn't recognize. This is general DNA. This shows that the region, this flexible linker is disordered. It actually has positive charges that help the Lac operon to move in a one-dimensional diffusion along the DNA. Then when it hits the correct binding site, the disordered region becomes a helix. I'm going to walk over, well, I'll have somebody, yeah. Yeah, okay. There's some helices. Okay, so that's disordered up there. But now go down to the lower picture. I can't see it because you're in the way. Okay. Now, there's a couple helices right at the top, at the very top. No, over to there, right there. Those come from the disorder. Okay, they switch into that helix. And when they do that, they bind in the minor groove, not the major groove. And they actually shove down in the minor groove, and that opens up the minor groove. And if you look at this DNA, it actually has a bend in it. Okay? And so that disordered region, everybody knows about the Lac repressor, but they're not taught that disorder plays a key role in the binding to the Lac repressor. That should be in every biochemistry textbook. Okay, shall we go to the next one? Okay, now here is a protein called P53. It's an extremely well-studied protein, and this is the line up and down is our predictor of disorder. If it's above the line, it's predicted to be disordered. If it's below the line, it's predicted to be structured. So you can see there's a central region that's predicted to be structured. That's the DNA binding domain of P53. Okay, now you have disordered tails. Look at all these disordered tails are bound to different partners. Okay, and this means that this is involved in all this different regulation that this transcription factor plays. Now let's go one more slide. Okay, and now here's a segment near the carboxy terminus, and this one segment binds to 4 different proteins. When it binds to one protein, it's a beta sheet. When it binds to another protein, it's an alpha helix. When it binds to another protein, it's a random coil. So this verifies the Pauling-Landsteiner hypothesis of 1939 and 40 that they fold into different structures and bind to different partners. Okay, I think that concludes my, so here's the summary. Sequence to structure to function. That's for proteins that carry out catalysis, for membrane transport, and proteins that bind to DNA, RNA, other proteins, and IDPs, and bind to molecules. And then sequence to IDP ensemble to function is involved in signaling and regulation. So that's the science part. Now, the Ralph Yount part. I didn't make any slides for Ralph Yount, but I'll just say a couple reasons. Ralph Yount's the reason I came to Washington State, okay? I interviewed, I saw the advertisement, I sent in an application, they decided to interview me, and so I came, and in my interaction with Ralph, that's the guy, I want to come here. But my wife, whom I love dearly, we've been married 60 years, she's from China, she's from Hong Kong, she grew up in Hong Kong. And the city of Hong Kong versus the city of Pullman was a bit much, okay? And so she said, I can't live there. And so I went back to, I was at Sloan Kettering at the time. She was at UMass Amherst studying physics at the time. And so we gave up the job. And so I kept trying to get a job. I couldn't get a job. And a year later, I saw in Science the WSU job was still open. So I called Ralph. And I said, can I apply again? He said, yes. So I applied again. And they offered it to me again. And so I came. Then he was most helpful all the way through, but he recommended to me that I apply to the American Heart Association for established investigatorship. Now, I didn't even know this existed, and I didn't think I had a chance, but he encouraged me, so I applied for it, and I got it, and it covered my salary for 5 years, which was absolutely instrumental to a young assistant professor to get extra time to do research and to get the lab going. So he really made my career. There's no doubt about that. And that's all I want to say.
So next we have another faculty recruit that I think Ralph was involved in, heavily involved in, and that's Mick Smerdon. I think one of the true, maybe first card-carrying biophysicist to join the faculty here at WSU. I can remember when he came, we were all celebrating that, and maybe Lin too. But anyway, Mick, it's great to have you here. Come on up. Oh, sorry, Keith. Okay. Advance your slides, okay? Yeah, okay. Okay. Yeah.
Well, as far as being a card-carrying biophysicist, I was actually really trained in solid-state physics until this was at Oregon State, and I went across from physics to chemistry when I wasn't drafted. I was on an S1 You know, I was not supposed to, not a student, but what did they call it when the draft was on? We were in Vietnam at the time, and so I got my degree in physics. This was in Minnesota, and then I went to the recruiter and said, you know, I'd like to get my master's in physics before going. At that time, I was going to go in the Navy. My father was in the Navy in World War II, and so that was kind of my dream at that time. And so I went to Oregon State and got my master's in physics, and then I took my physical, and it's the one exam I failed because I had already started, I have MS, multiple sclerosis. And so apparently it was obvious in the physical. And so on the bus trip back, was really kind of befuddled. I was planning on going to wherever the Naval Institute is. And so then I decided to join this field called biophysics because they had just started a program there, and they had some really excellent faculty. Ken Van Holt was one. Irv Eisenberg was the person I went to train with. Anyway, so I learned a little bit about then physical chemistry and the study of proteins. Keith and I actually taught some courses together, and we discussed what are called histones, proteins that bind DNA in what's called chromatin, okay? And after some of our courses together, we would talk about, even back then, disorder, and the one histone that I had studied in graduate school, which was histone H1. And that's a candidate that probably enters into that 50% or so number from human proteins, because there's a lot of histones in the cell. Anyway, when I came, I felt like I was really, if people asked my label, it was really physics with a small bio in the footnote, because I felt really somewhat out of place when I moved here. And so that gets me to the fellow who we are celebrating today. We've heard so many adjectives today about Ralph. I don't cry very easily, but it would make me, when I thought about it, it was really going back in time to that time, just over the last few days. The reflections made me think of how important that fellow was. And like so many of us have already talked about. And again, I wasn't a student. A lot of you were. I was like Lin and Mike, a young faculty member. And again, he also attracted me. At the time, I was looking very seriously at another school that had a medical school, well-known school, on the East Coast. And I, at the time, was thinking maybe I should go to a place that had a good, had a department that had, say, an MS Institute, for example, at the time. I didn't know. At the time, I didn't know how fast this was going to progress. And so Ralph and I talked as much about human stories as we ever did about science. Even though he was very structurally minded, I guess I never thought of Ralph as a biophysicist. I always thought of him as a chemist. I mean, if there's one term I learned from Ralph, it's phosphodiester bonds, you know. And we did have some, actually, what I used to call fireside chats, and I don't know if Lynn and Jerry had these, or Mike, any of you that kind of came around the same time, but our lab was right across from the Yount lab in Fulmer Hall. And I had to have it all remodeled because at that time, believe it or not, I was doing cell culture. I mean, that was about the strangest thing for a person trained in physics as I could think of. Fortunately, my postdoc advisor at Washington University, his name was Mike Lieberman, he sat with me. I mean, first thing he gave me was HeLa cells, and he said, Mick, you just keep trying, and if you can't get these to grow in a couple weeks, you're right, you don't belong. Unfortunately, I was able to get these cells to grow. Well, once I came, Ralph would, I guess it's because we tended to both stay late in the unit. At that time, I was doing my own experiments. I had just a couple people in the lab. And sometimes I just couldn't get to them until the end of the day. So Ralph and I would be in there rather late sometimes. And, you know, in the evening, we would go home. If I saw him, you know, goodbye, Ralph, et cetera. Once in a while, he'd say, you got a minute, Mick? And so this was the beginning of what I called fireside chats. And this would happen maybe once or twice a month. And so initially, he never told me how to do my experiments. I mean, I studied DNA repair, and just the fact that it had DNA in there, I think made him, you know, cringe. But it was something he actually asked me about and well, I won't get into this because it'd take too long, but he actually impressed me by the fact that he seemingly was interested in some of the things that we would talk about that I knew, because he would ask me basic questions, and I loved that. I used to tell him, I said, Ralph, there's one thing I don't believe in, and that's something called a stupid question. There's no such thing in science, in my opinion. Some stupid questions have led to discoveries. So at any rate, Ralph and I would have these chats, and during those chats, I learned about his favorite molecule, which of course is ATP. We've heard a lot about it. I learned a lot about it. And my interest in his work was just from the get-go. I always wondered how chemical energy was transformed into mechanical energy. And there's two really, I think, very visual components of a cell where motion is tied to ATP hydrolysis. And clearly muscle is and I would have actually gone into it probably if a position had opened for a postdoc in that field, okay? It just so happened that this position in St. Louis opened up, and he was studying DNA repair. And the other system is, of course, chromatin. Chromatin is as dynamic a structure in the cell as you can pretty much think of. You know, nowadays we talk about the relationship not of gene sequence and so on, but so-called epigenetics, and how the chromatin is decorated, how that influences what proteins bind or don't bind, how loose the chromatin becomes or remodeled, And a lot of that relates to repair, okay? You saw a lot of examples in Keith's slides when he put them up here with P53. There was some BRCA proteins and so on. A number of these proteins turned out to be chromatin remodelers, okay? And when I was a graduate student, a postdoc, in St. Louis, this is back in the early 70s, I was fortunate enough to find out that chromatin remodeling occurs every time there's a DNA repair event. And that was actually the first time that was ever shown. And it turned out to be kind of predictive of what we now know is a very functioning process that occurs for a number of different mechanisms in the nucleus. And we now call it chromatin remodeling, and there's a number of chromatin remodelers. By the way, a number of these remodelers tend to have mutations that end up in things like cancer. Okay. So anyway, during these fireside chats, the next, and by the way, I was going to say, the title that I have, I figured by this time that we would have used and heard every adjective to describe Ralph that I could ever possibly think of. So instead of adjectives, I gave acronyms of things that I thought reminded me of Ralph. And so ATP, of course. During those early days, well, initially when I got here, Jim Wells he had just finished his thesis defense and was still here. And so I heard a lot about, next slide, please, this thing, ATP trapping. And it really, oh, and by the way, Jim, I'm sorry I put you on the dog that's sleeping, but it reminded me that when I met Ralph, he had this student that he told me about that was sleeping in his truck when I first got here, and so, or when he first came. And so there were only 3 dogs, so that's why there's only 3 names. But Ralph and I discussed the trappers and so on. Oh, that's my cane, I'm sorry. It's really put in here to wake you up. So one thing that this led to is he had a student, and I don't know where he's at right now. His name was, last name was Munson. It was a Keith Munson, I think. Yeah, I don't know if he was a pre-med or what, but he was very interested in determining the structure of the cross-linked subfragment 1, so the myosin SF1 portion that Ralph basically spent much of his career studying. And so if we can have the next slide, please. We entered into one scientifically collaborative project that I was involved in with Ralph and his group. And what impressed me during the time is that, yeah, Ralph knew a number of things about like fluorescence and so on, but he really was, he wanted to learn more about this AUC, analytical ultracentrifugation, which Lin has already mentioned. So I picked AUC as another acronym to describe my time with Ralph. And again, during these kind of random fireside sessions, I was impressed. And in fact, to the point that I decided that it restored my belief that old dogs can learn new tricks. And I can't use any more adjectives than have been used for this fellow, but it's just another part that amazed me about him. And so at any rate, this was just the study, and if you could press it again. So it was very simple. Now you got to remember, this is back in the like 1982 or 81, whatever. We didn't have crystal structures and so on of the cross-linked protein. We were just asking, Keith and Ralph were wondering, well, what's the general shape of the cross? Where were these cross-links occurring? Because there was just one ATP binding site per subfragment, and was it linked end for end to make essentially a prolate ellipse, or was it more spherical, binding more in the middle? And this is something that we could do with the techniques of analytical ultracentrifugation. So what we did is we combined what's called sedimentation equilibrium with sedimentation velocity. And the bottom line is, I won't go into detail, but if next, you can just push it. So it turned out that the structure was clearly more spherical in the cross-link state. And so that there wasn't, and this told Ralph, you know, where the ATP binding sites were. A little, and again, we're talking, we didn't have cryo-EM, and so on and so forth, like you do today. And so that was just one, again, for me, a part that I reflected on where Ralph really proved his worth to me. Just those sessions, because it was one-on-one, and he didn't have to do that. And so finally, just one other thing that came out of those talks. Next slide. All right. So one time he asked me how, so we had now 3 children, my wife and I. My wife, I don't know if she's here right now, but we're celebrating our 55th anniversary. Yeah, I'll say we've fought kidney cancer with her for the last 5 years, 4 years, and I can just thankfully say that she was supposed to be gone 2 years ago when they first diagnosed it. And a lot of what we've heard about today, Mickey, I mean, it's incredible how important the new treatments have been, in this case with monoclonal antibodies, that my wife is still alive. And so that's an aside, and I apologize. So at the time, we had 3 children. Here you see my oldest boy. I don't know how old he is. I don't know, 7, 6, 8. His name is Jason, but that's how we camped when we first came here. The kids wanted to camp, and so we got a little 9-by-9 tent. And the 5 of us would stay in this tent when we went camping. And you can imagine what that might be like. And so Ralph and I talked about it. I can't even remember how it came up, but immediately he said, Mick, I got something for you that's a solution. So the next slide. He had this Coleman pop-up tent trailer. And he said, Mick, we don't use it so much anymore. Take it whenever you want. We went to Banff and Lake Louise. We went to a number of places throughout the Northwest here in that tent of Ralph's, and it was so much nicer. It even had a little kitchen you could cook in, and with 3 kids, it just made all the difference. So I can end by what my wife used to say about Ralph. If you just hit it one more time. He was a big teddy bear. And to our family, he was so many more things than I had ever expected. And I am so glad that I never went to the other university that had the medical school. All right, thank you. Now,
All right, I think next we have one of my best friends and ex-roommates in graduate school, Gary Pielak, who, like Mickey Urdea, worked in a lab that we had joint lab meetings, Ivan Legg's lab, And but when the weekend came and the sun went down, our house turned into Animal House. And Gary will tell you a little bit about it. Thank you, Gary.
Thanks, Jim. So we've heard about Ralph making phone calls, but this one was unique. He didn't call me. Now, I cannot figure out, I might have put the wrong number on the application, or he might have called and I said I wouldn't be there. Here's my number at home. Ralph called my mother. And so my talk is in 3 parts. What did Ralph mean to me? 2 discoveries, and how WSU prepared me for what I'm doing. So there's my mother and father, and they're standing at my house, at the house here on Maple Street in Pullman when they came to visit me. Yeah, so when I came home from Bradley University one time, my mother said, this chap named Ralph Yount called me. We were on the phone for an hour. Now, my mother never finished high school. And so what they talked about, I don't know, but Ralph could charm the birds off the trees. And my mother basically beat me up until I decided to come here. And just to get it out of the way, that's me in graduate school down there in the corner. Oh, I got to go back. And, Ralph was like your father or your very wise uncle. But I earned my PhD with Ivan Legg, who was like your crazy uncle. And here is Ivan with another crazy uncle, Carl Poole, out camping. And I remember this first summer, I wondered what the heck I had gotten myself into. And here, and luckily always had Ralph to go back to, because Ralph was, really solid, and he was very wise. And then you'd go back to Ivan, who had a million ideas. Okay, so what have I done since I left Pullman? I went on to do a postdoc at the University of British Columbia, and then I did a second where I learned site-directed mutagenesis. Then I went on to Oxford, where I learned some NMR. Then in 1989, I joined the University of North Carolina, and I've never left. Okay, so two discoveries. I'm going to talk about the 2 projects my lab works on. One is macromolecular crowding, and the other is the ultimate crowding. That is, you dry the protein out where there's no water left. So this is a simulation of the inside of a bacterial cell. Let me back up a second. If we look at higher eukaryotic cells, the concentration of macromolecules, mostly proteins, is about 200 grams per liter. In bacterial cells like this, the concentration of protein is about 300 grams per liter. Mitochondria are about 400 grams per liter. So what the heck does that mean? If you had an egg white this morning, that's only 100 grams per liter. So there's a lot of space, 30, maybe more, 30% of the space in cells that's taken up by proteins. But most of what we know about proteins comes from dilute protein in buffered solution. So when I started on this, I earned tenure on the biophysics of a protein called cytochrome C. So after I earned tenure, I said, I want to work on a problem that's impossible. That's very difficult. So I said, okay, I'm going to figure out what happens under crowded conditions. So the idea at the time was very simple. It's called the elevator analogy. So what do you need to know? You need to know that proteins have 2 states, the Chris-Anfinsen idea, where you have a more compact folded state in equilibrium with a less compact state. And you know, the physical chemistry of this is really simple. Even I understand that. So in dilute buffer, it's like being in an elevator all by yourself. You can be expanded. You can be compact. It doesn't matter. But inside a cell where it's crowded, you can't be like this anymore. You've got to be compact. And so under crowded conditions, that dead French guy, Le Chatelier, would say that you've got to push that equilibrium towards the left, towards the compact state, which means that crowding should stabilize proteins. And you notice I didn't mention any chemistry at all. In other words, it's just the proteins can't interpenetrate each other. So there's no chemistry, and so this is an entirely entropic stabilization. This was state of the art. And I initially set out to just prove this was true. Now, I see there are some graduate students here. And you should probably hide your eyes, because I'm going to go over, I think, 6 PhD dissertations in one slide. So the first thing we did is we figured out how to measure that equilibrium inside E. coli cells. And the protein was the protein is in general less stable than it is in buffer. We started out by the first things we looked at, we couldn't make this idea work. And then we did something else. We said, well, maybe it's like some kind of simple chemical interaction, but if we crowd it some more, the protein will become more stable. So we osmotically shocked the cell, so we sucked the water out of it, so we made it even more crowded. Stability goes down even more. So then we said, well, bacterial cells are more crowded than oocytes, so let's make the measure in oocytes. So even though they're less crowded, we get the same destabilization. Then we looked at protein complexes in cells. The complexes, this is a chemical reaction where you have two things coming to one. It's more stable. So what the heck is going on? And we also knew that even being in cells could change the reaction. We had this system where in buffer, in the test tube, it went from a folded monomer to a folded dimer. But in cells, it now went from an unfolded monomer to a folded dimer. And most importantly, we took the time to measure the temperature dependence so that we could get the Van 't Hoff enthalpy. Almost every system we looked at, there was an enthalpic effect, but simple theory says there should be no enthalpic effect. So I wish I had time to put you all to sleep with the theory, but I don't. See this is a very crowded situation here, but they all have floaties on. And those floaties are the chemical interactions that make all this possible. Now, it's not this simple. It's more complicated than this. But what it comes down to is that the initial theory ignored all the chemistry, and there's a lot of chemistry going on. Okay, that's story number one. Now, I'm not finished with this. I took 4 graduate students last year, and I'm going to finish this project. We have a model that really works, and it works on simple polymers, and we need to test it with proteins. And that's what we're doing now. Okay, now, the ultimate crowded condition is a dry protein. And the way I get into this is, I don't know, a decade ago, some postdoc from biology comes into my lab and says, I've cloned the tardigrade proteins that let them survive desiccation. So I said, what the hell's a tardigrade? And tardigrades, they're finally known as water bears. You can dry them out, put them on the shelf for 20 years, add water, and they come back to life. So we did the physical chemistry on these proteins, and I got to tell you, I've worked on some weird proteins in my life. This is the weirdest one. Here, once you purify this, we put it in an Eppendorf tube, and you can see that it's a beautiful clear gel. So it's a disordered protein, but it's a disordered protein that forms a gel. This was really great, but I wanted to figure out how to get some grant money out of it. Because, you know, let's face it, tardigrades, they don't have health problems. But if you look at biologic drugs, they're quite labil. You can't heat them. You even shake them. You agitate them and they denature. They have all kinds of covalent reactions. You change the pH, they die. So we have to keep them cold. And this is called the cold chain. And I don't have time to go into it, but let's just say this costs an awful lot of money. So we thought about, well, what if you could dry these proteins out and store them dry so you didn't have to refrigerate them anymore? And this was how we got in to studying dry proteins. Now, we had to invent a whole bunch of technology to do this, but I'll tell you about one of the discoveries. We looked, often when you formulate proteins, often you use sugars. And there's one sugar that everybody uses. It's called trehalose. And so we wondered, you know, and I'm a chemist, I looked at it, trehalose doesn't seem very magical to me. So we used these techniques we developed to look at dry protein stability in glucose, sucrose, maltose, and trehalose. And what we found is that they all do the same thing. They all increase intraprotein H bond strength. And they probably work by the OH groups on the sugars, replacing the OH groups you've sucked away from water. So what the heck is magic about trehalose? And it turns out we didn't even have to do any experiments to figure that out. Now, trehalose, we all know trehalose because if you've had sea monkeys, the reason that brine shrimp, the reason you can have sea monkeys is as you dry them out, they make buku trehalose. And this protects them, so you can just add water and they start swimming around again. And after we couldn't figure out what made trehalose magic, we finally did something that nobody ever does. We read the literature. And it turns out that physical organic chemists in the 1950s showed that kinetically, trehalose is far and away more stable than any other disaccharide, kinetically stable, and that's why nature uses them. All sugars protect the same, but trehalose will be around for an awfully long time. Okay, so that's all I have that's my two stories. So I've got to acknowledge, you know, I've had 50 graduate students. And if it hadn't been for Ralph calling my mother, I might not have had any. So that's my current group. And then I have to acknowledge my collaborators in Israel. They've forgotten more physical chemistry than I'll never, ever know. So the Pielak lab, we make these weird crowding experiments, and they help us interpret them. And I've got to thank the National Science Foundation for giving me a lot of money, and the NIH, and the Keck Foundation, and the U.S.-Israel Binational Foundation. What would happen if Ralph hadn't called my mother? And so how did WSU prepare me? Excellent, excellent classes from Yount, Dunker, Courteau, and the scariest one of all, P.E. Kalatakuti. This man scared me. And I know, Mike, I didn't mention you because I had never had a class. I took a bunch of exams, but never had a class. And so what did I learn from Ivan? Well, I learned a lot of things I shouldn't have learned. But one thing he said that just comes back to me on students say, why should I finish my PhD? And I think a lot of students, they say, well, I've learned NMR. I'm going to do NMR as a postdoc and forever. No. Your PhD proves it's your union card. It proves you can start and finish a product. We need to keep that in mind. Now, one great thing about Pullman, there were very few distractions. But you could go 50 miles in any direction and be eaten by a bear. That was a lot of fun. And the other thing that I learned is I have much less fear of volcanoes. And that's in front of Fulmer, collecting some ash to see if it was going to kill us or not. And I learned a lot more. But let's talk about that over a couple of drinks. So that's all I have to say, but I've ran a couple of few seconds of pictures here. Here, Jim, you can go to the next person.
All right, the penultimate is Jerry Hazelbauer, who's going to come and talk. And he's currently at University of Missouri, but was here with Lin Randall before, and they were two amazing PIs here that made so much difference to the department, and Ralph was absolutely involved in recruiting both of them. Jerry, I think, was also chair of the department for a while.
Okay. Make that full screen. So I'm going to talk about some of the same things you've heard from a slightly different perspective. So WSU in protein chemistry, which we've heard quite a bit about, Ralph and me. Well, as Lin already mentioned, she and I arrived at WSU. She arrived as an untenured associate professor because she'd never been in the American system as a faculty member before, so that's why she was not yet tenured. I was a spousal accommodation. I came as an associate scientist, which meant not only did I not have tenure, but I wasn't on a tenure track. Ralph encouraged me, and the way he did that was not by saying anything explicit, but by treating me like any other junior faculty member. There was no assumption that I was different or that I was going to have to go learn some other area, but that I was going to do fine. And in retrospect, that meant a whole lot. Now, it also happens that having been a spousal accommodation, I was chair of one or another departments for 25 years, and you have to learn about spousal accommodation. And I could always start by saying, look, I know, I was one. So I want to talk about one thing that hadn't been mentioned only in passing, which is protein chemistry and the NIH biotechnology training program. So in 1988, a few years after Lin and I arrived, we got to a situation at WSU where there were no research-based NIH training grants. There was a clinical one or two in the vet school. There had been ones in the past, but as things got tighter, money got tighter and tighter, those grants weren't renewed. And so we didn't have one. And it's very hard to get into a existing training grant program because the current institutions do everything they can to get it renewed. But what happened was a new training grant program, first one in a long time, was announced by the NIH in biotechnology. And a group of faculty got together to discuss this and said, okay, maybe we should make an application for this. We've got a better chance with a new program than we do with trying to break into an existing one. So we had a lot of discussions on what was our strengths. And it wasn't gene manipulation. We were not close to having people who really were at the cutting edge there. But
protein chemistry was a strength. And we could point to people, including some of the speakers today, of, well, you know, we already trained some people who did pretty well and are doing pretty well. And you've already heard the list of, I would say, almost intimidating faculty who did protein chemistry as a life work and trained people to do that. So most of the applicants, I later learned, for various institutions around the country, really emphasized gene manipulation, and site-specific mutagenesis was very new at the time. We were the only ones who said, well, what we're going to do is train people in protein chemistry so they know what in the world they're trying to produce. And Ralph was absolutely central to that. And you've heard the list of the other faculty. And it turns out we got it. I got prevailed upon to coordinate that application. And then when we had to put it in with a prospective director, I got named for that, and we got it. We were one of 9. We were the only institution not on a coast that got one of those. 36 years later, WSU still has that training grant with an emphasis in protein chemistry. None of the other 9 are still funded. So something happened right, and it's persisted over the years. And it was really Ralph that helped in a very important way drive that. Ralph was also, as a colleague, was really a mentor and a friend, and Mick Smerdon touched on that in a very effective way. So he was always ready to talk, as Mick said, about science, about mine, as well as his. And so I pretended I knew more chemistry than I know, and listened to him talk about his nucleotide analogs. And he pretended he was interested in transmembrane signaling when I talked to him, because I worked on bacterial chemoreceptors. And, you know, as we all have had, there are times in our interactions with graduate students that we didn't know how to handle things or what to do. And Ralph was one of those people you didn't even have to hesitate. You could go into his office and say, "Can I talk?" Plop down in the chair he had that was kind of too soft and fell over and fell in the middle. And he'd listen to whatever issue you had with students, with staff, with other faculty members. And he'd come up with some impressively wise comment that got you started in the right direction. So that was really being a mentor in a way that was extraordinarily effective. And then to talk about the department and its faculty, and it was particularly once I became chair. And there were some challenges now and again with some very talented and very independent faculty, and a need to somehow get some things done and get direction in the department and decide how we're going to do what we wanted to do. And again, Ralph would listen and come up with some comment that helped me along the way. And he'd also talk about politics, and we won't go into that here, but he was passionate about that also. One thing that really hasn't been mentioned very much is Ralph in national service. I mean, this is a man who was a superb teacher, an extraordinarily successful researcher, and he gave his time to national service in multiple ways. So I can remember the second time he stepped down after 5 years in the editorial board at JBC, and the lab, and we were across the hall, had a party for the rest of everyone in the lab, because not so much for Ralph, but that Ralph wouldn't have to deal with all those papers coming in all the time. And he, of course, took them all very seriously. And then he was president of the Pacific Coast Conference early on, and then a series of responsibilities, starting in the Biophysical Society. He was first on the council, then he was president-elect, then he was president of the Biophysical Society. From there, he started serving on FASEB, the Federation for American Societies of Experimental Biology. First as a board member. It was a time it was quite common for the past president of a society that was a member of FASEB to have that past president now be a representative, to be on the FASEB board. And that's what Ralph did. And the president of FASEB is chosen by the other board members. It's a nomination and voting process. And not surprisingly, Ralph was strongly elected to be president of FASEB. He's somebody you trust. He was somebody who you knew could spend the time, because he'd take the responsibility seriously, and be an effective spokesperson. Now it happens in the years that he got elected, when he got elected, that was when there was a major push in Washington to double the NIH budget. And Ralph, as president of FASEB for that very crucial year, was central to that effort. And I was chair at the time here in biochemistry, and I remember talking to him about reducing his teaching load. He, of course, said no, and I said that wasn't an answer he could give, because literally he was flying to DC every 2 weeks to meet with various influential people. And it happens one particular trip, he and I shared a flight together, because I was on working in proteins and the protein science. And I said, Ralph, how are things going? He said, well, you know, we're meeting with members of Congress, we're meeting with their staffs, we're trying to get support for the real push here. And he said, just recently, I perfected the elevator speech. Our political operatives at FASEB, and there's some very, very good ones, said, Ralph, you gotta have the elevator speech. You gotta be able to give it in those 10 to 15 seconds while the elevator is moving up with somebody who needs to hear what you wanna say. I said, okay, Ralph, what's the speech? He says, it's simple, Jerry. You walk into a congressional office and you meet sometimes the member of Congress, often a senior staff member, and you look that person in the eye, and you know it's this Ralph. He's going to be good at looking you in the eye, and say, you're old enough to know you're going to die. And you're smart enough to know that before you die, you're going to hurt. The NIH can help. End of elevator speech. They doubled the NIH budget. So I leave you with Ralph. From all the other things he did, he gave service to the whole community. And he did it without any begrudgingness at all. It was because it was something you ought to do. Thank you.
All right. Well, these talks have been a survey of Ralph and all of the folks that have been affected by him, all the ways that he's launched the careers of young faculty and students. This is really an amazing. So I'm so honored that you guys are here, and I'll tell you a little bit about my arc here with Ralph. So I'm going to tell you stories of protein engineering, which is really the field that I'm in, that Ralph inspired in me. And I want to start here. Well, like Gary, the first time I talked to Ralph was after my mom had called me in Lake Tahoe, where I was a ski bum trying to figure out what I wanted to do. In parallel, applying to medical school and to graduate schools, not getting into any until this man, Ralph Yount, my mom said, "You got to talk to him. He's this might be your ticket." And so from a phone booth on a snowy morning in Tahoma, California, Ralph called to say, "Hey, we're going to admit you to WSU." I said, "Oh, I can't believe it, really?" And like Gary, he kind of sweet-talked me into WSU. And like Mickey, I too thought that we were in the evergreen state. Oh, that means there's all kinds of trees around and everything else. Because we only looked at the catalog of WSU where they have those things, mostly of those pictures from Moscow, Idaho. So Carol, my wife to be, we came up to, drove up to Washington State, and then we got through all of the beautiful, evergreens and whatnot into the wheat desert here. We're coming from Berkeley, by the way, where I was an undergrad, and culturally that was just a real shock. But then I met Ralph, and like others have described him as a teddy bear. Actually, students described him as a teddy bear with fangs. Some did. He was, so he kind of rescued me. I felt like a rescue dog, And to me, it was just I learned so much from him. As Mick Smerdon mentioned, we discovered together this phenomenon of disulfide trapping. I worked on a really mysterious inhibitor we called Biomud. Kei Nakamai may remember that. And it had, it was the first thing we tried to do is to purify the thing. And when we purified it, it was just a bunch of different fractions, metal complexes and whatnot, which is why I interacted a lot with Mickey and Gary and Ivan Legg. Ultimately, we fractionated it and figured out that all the activity went away. What's happening? And then we got the idea that, well, maybe the activity is a combination of some of these things. And we did add-back experiments, and lo and behold, we could recover activity. And that activity was this metal complex called cobalt phenanthroline, accelerated in its reactivity with ATP, that we found in the end was due to disulfide, was due to thiol chelation and trapping of ATP at the active site. And so I learned so much from that. It was just incredible. And so after I then did a postdoc at Stanford with George Stark. And after that, I was so lucky to come in to Ralph. I want to acknowledge two other people who really influenced my career too. One was Mickey, and Mickey, I just fallen in love with chemical modification of proteins. Mickey pointed out a very important paper by Mike Smith on this new technology of site-directed mutagenesis, which is being applied to DNA control regions at that time. And we said, oh my God, this could be used to much better than chemical modification. And we could just mutate any protein we wanted by, changing the DNA sequence and then expressing the protein. Of course, that had complexities, as you heard from Bryan Lawlis here. When I was trying to figure out what to do, I wasn't going to get a job doing that, in an academic job, because it required oligonucleotides, required expression systems, it required cloning, it required sequencing. I'd done none of that. And then my other graduate friend, Bryan Lawlis, I was talking to him about this, said, Genentech's trying to figure out what to do with the recombinant DNA technology. They just might be interested in an experiment where you see, could you make a protein better by site-directed mutagenesis? And so one thing led to another, and I went up there, interviewed, and I was amazed. Genentech was a bunch of postdocs and no adult supervision. Much like Chiron was, where Mickey was, because there's no way, no managers that understood enough about recombinant DNA technology that they could figure out, would any products ever come of this shit? So anyway, so we took the approach of, let's pick a model toy system, and I did with my collaborator here, Dave Estelle, also at Genentech. This enzyme cytolysin, simple simple bacterial serine protease secreted from bacillus. It's a scavenger protease. We knew its structure, determined by Joe Kraut. We also knew its mechanism, and we had some ideas where we might want to poke and prod it by site-directed mutagenesis. So we started doing that with the view that could we make this enzyme better? Okay, that was our task. And one of the first things we saw was, this enzyme is very sensitive to oxidation. Where at this particular methionine 222 that Bryan mentioned. So that was our first target. That one. Indeed, we could change that methionine to a number of other things. A completely conserved methionine, conserved in all cytolysins, but it allowed us to change that and made it tremendously oxidatively stable. We went around, made it stable in other ways with disulfides, other mutational changes. We altered its specificity. And then this guy from the business development group came down to tell us, hey, that toy enzyme that you've been playing with that you could improve, that's the largest industrial enzyme market. What? That wasn't our intent. And that led to the founding of to this product that Bryan mentioned, in the founding of Genencor, with the motto, by the way, if you can't truck it, F it. So that was an amazing time. Genentech was also transitioning, trying to figure out what to do with recombinant DNA technology. And Dave Martin, who is now hired, the adult in the room, was hired to be director, came down to say, Jim, well, that's cool that you're doing this stuff on this industrial enzyme, but that's not us. We're about this. We're about pharmaceuticals. And so he asked, could we find out how to make their first product, human growth hormone, work, make it better, even understand how it worked, because they were selling it for short-statured kids. They didn't even know how it worked. Okay, so enter my cohort in crime in protein engineering, this department that we formed as a result of the cytolysin work and other things. And with Tony in the structural group, we solved the structure of human growth hormone, showing that it actually had a really interesting mechanism of action. One hormone bound two identical receptors using two separate sites on the molecule. And with Germain Fu, we were able to determine that this dimerization mechanism, first time it was observed structurally in a hormone receptor, actually is critical for its activity. And from there, we were able to actually make mutations in growth hormone that would allow it not to just be an agonist for kids, but also a useful therapy for people with acromegaly. This is a fatal cancer that's not treated with drugs. Can be treated with surgery once to remove the cancer from the pituitary, only successful in half the people. And you can only do that once. So we made a drug, an antagonist of growth hormone that then got approved in 2003, now sold by Pfizer, called Somavert, and it's first-line therapy for acromegaly. One of the things we discovered in this process of understanding how the hormone bound to its receptor was through mutational analysis we called alanine scanning, that only a small set of residues at the contact interface are actually critical in binding. It's amazing. A structural biologist would look at the contacts and say, everything's important, you know? That's like a Catholic view of sperm. So it's not all important. There's a small critical set in the middle, and in fact, it generalizes not just a growth hormone receptor interface, these other interfaces as well. In fact, there's been 50 interfaces, which this has been shown. This notion of hotspots. Now, why is that important? Why were we interested? We wanted to be able to actually make small molecules that could mimic these large hormones, okay? Could we actually do that? Well, hotspots suggest you might be able to do that. And that led to the founding of this company called Synesis. This is the group that joined, a bunch of them from my lab at Genentech. There's also a co-founder, John Ellman, who was from Berkeley in that picture. And we were starting a company is crazy. You know, when you start a company, it's just you and the world in a gas station, and you know, and it's always building the ship while you're sailing. And I like this pirate ship kind of notion. That's what I consider my lab, actually, is pirates stealing and having fun from nature. So in this process, what goes around comes around, because the key technology at Synesis was a covalent drug discovery notion, because there weren't starting points for protein-protein interface inhibitors. There's nothing in the literature. There's no substrates. It's not like building an inhibitor to an enzyme where you know the substrate or anything to go on. In fact, there's not high-throughput screening hits from them even. So we developed this site-directed way using cysteine chemistry that I learned from Ralph, a technology called tethering. And what we had was a library of the, see, you can see on the left that balloon with the SH coming out of it. That's meant to be a protein with a thiol near an active site we wanted to, or a binding site we wanted to interrogate. The balloons over that arrow are a library of disulfide exchangeable compounds that we built, about 2,000 of them. And then we just mix them with the protein, and by thiol disulfide interchange, the best one wins. So we can detect that by mass spectrometry. So from that, I'm just showing one example of this. For instance, on IL-2, that was of interest to us. We engineered a bunch of cysteines in and around where we knew the binding site to be. So you can see the blue helical model with those yellow dots. Each of those yellow dots is a position we put a cysteine. Then we found a bunch of fragments that could bind to those cysteines, and then chemists were able to link those together based on their positioning and their chemical content to ultimately create in the bottom left of that a small molecule, first small molecule to actually bind a protein-protein interface that broke the micromolar barrier and with the structure defined. So that led on to a whole bunch of other collaborations and studies. And in fact, in the end, there were 2 drugs that came out of Synesis that I'm very proud of that we ended up licensing to other companies. And we continued, when I left Synesis in 2008 because I was missing inventing and being among students and postdocs that I really loved, and I had them all along the way with me. To be looking for not only protein-protein inhibitors, but also allosteric site inhibitors too, because those are also rare to find and hard to find. But if you have a sense where they may be, you can use tethering to put thiols in and around them. And one of the most famed examples of that was this collaboration I'll just mention quickly between Kayvon Shokat's lab and a really good student he had, John Ostrom. They were interested in finding out, could we find inhibitors to KRAS? KRAS is present in 30% of tumors. It's probably the most important oncogene. And so they were trying to build covalent modifiers that would go in and displace the GTP in KRAS, but that binds with a 10 picomolar binding constant. You're not getting it out of there, dude. So turns out that there was a cysteine variant in many of these oncogenic KRAS, and I suggested in a thesis meeting with John, hey, why don't you use our tethering library and see what you get? So they did, and they found these compounds. Shown here are the tether hits that they found. Now, these fragments can be advanced by talented medicinal chemists, and because it's KRAS, there's going to be interest. So that was the case, and in fact, a bunch of pharmas, and little pharmas and big, jumped on this. There are now 3 approved drugs for KRAS cancers, lung cancers. It's a super important. There are 29 current clinical trials going on with this. Okay, the last little chapter I wanted to say about the work that we're doing currently is I'm very interested in how the cell surface proteome, 5,000 proteins that encode it, change from health and disease, because that's where all the biologics bind, all the antibodies bind to those targets, half the small molecules bind there too. So I'm very interested in interactomes at the cell surface. Now, we know a lot about those inside the cell, but we don't know much about them on the cell surface, because for cell surface proteins, in a protein in the cell surface, and Gary will relate to this, it's you lose 3 of your 6 degrees of freedom. You can only move in 2 dimensions. You can only rotate in 1 dimension, the axis of the protein itself. So while they're very important, we know much less about them, which is why you see, if you look in, like, if you go on Google and you ask, give me the signaling pathway for growth hormone, something we worked on, you'll see them acknowledge the dimer we found. But there's nothing else around it except the ignition of a whole bunch of complexes that happen inside the cell. All the stuff on the lateral side, on the membrane, we're blind to, because these complexes are so fragile to hold. So I've been involved in this technology we call molecular photography. And there's actually a whole community that's involved in this kind of stuff. And what we do is we put, it's a molecular photography is where you put a photocatalyst on your target of interest, your membrane protein of interest. And then what you do is if you flash light on it of the right wavelength, you can ignite different highly reactive radical intermediates. And this one here that we chose is with ESNY, which is one of the most active photocatalysts in the garment industry, because it'll induce polymerization of acrylamides and things like that. But it'll trigger either this aryl diazerine down below, or this aryl azide, which, by the way, we know about from Ralph's lab. We worked on that. Or these phenoxy radicals. They have different half-lives, and when they're triggered, each of them has a biotin on it. When you flash light, you ignite the photoprobe, then the photoprobe only has a certain range, because its lifetime is determined by the radical itself. So they have different distances that they'll fly. So it's kind of like a Google Maps of molecular labeling. So photolyze this target which has it there. And this has been, I'm just going to show you this quickly. This is a study we just have recently, is coming out soon, on EGFR, where we've been able to trigger the identity of different partners as time goes on. So what you do is, on the left, no EGF there. We can find out what that neighborhood looks like. Those proteins that are shown there on the far left are proteins that are within about 150 angstroms of EGFR. When you add EGF, boom, there's a bunch of changes that happen. All those blue proteins in the middle associate. So some of the ones on the left leave, some new ones come in. And then on the right, after 60 minutes of signaling, it ignites a bunch of transcription factors, and we see those too. So these are going to find, I think, new neighbors for important drug targets and allow us the opportunity that we can build, for instance, bispecific antibodies for them and the like. So, so that's the science part of this. I just want to go back to this thing here, and thanks for the inspirations. And these are 4 of the students, and all of them made a big difference in our life. You know, as PIs, that while we PIs try to impart our wisdom onto our students, actually much of it is done by the students themselves. And the sharing of information and that's so important, which is why having a really good lab culture is critical. You want your students to be engaged with each other. You don't want them competing with each other. You want them to be supported and know you've got their back. And up in the upper right is one of our lab pictures showing us together. And of course, Ferdinand's, which I sampled a little bit of their milkshake today. Ross and I went over. And of course, Ralph, who was he was the ringleader. He was at the center of this. For me, it was a lifelong inspiration. We passed our IC50, as I call it. I've known him longer than I haven't. and I saw him first talked to him in 1974, and I was so lucky to have seen him on his, when he was 92, came up and visited him. And it was just special. Like everyone's talked about here, you know, when you're anxious or anything, I'd always call Ralph. He's always available. And within 10 minutes, I would be going from hair on fire to relaxed. And he was just that kind of calming influence of like, steady, man, take it easy, dude. You know? And so he was an amazing guy. And then he even picked me up at the airport, that was great. So tremendous remembrance of him. So I'm going to close here, but I just want to acknowledge the organizers. I want to thank Claire and Abby and Darby and Amy for assembling us today. It's really been great. And lastly, before we go, wait, one more. I want to acknowledge Allison Yount, who's here. So half of Ralph is here. And so it's really great you could be here with us too. So with that, thank you all so much for being here. We'll have some sort of a reception, I think. Okay. I wanted to say, as these pictures are showing, we have an open opportunity for an open mic for anybody who knew Ralph, aware of him and his influence, and what he might have meant to you. So what I'll do is I'll take this microphone, and if you raise your hand and want to say a few words, and in fact, I might start off by nominating some of the folks that didn't talk today, but I think knew Ralph. And I'm going to start with Scott Braxton back here. Here you go. You don't have to get up. You can do it in situ, however you want.
So Ralph also called my mother. And my mother said, "This is a really nice guy. You need to talk to him." There were only 3 places I was considering going for grad school. Tom Check at Texas, Boulder, and here. And before coming here, I was going to go to Boulder, Colorado. But when I came here and talked to Ralph, it was clear this is the place to come. He had such a personable characteristic. He told me he spent his Saturdays and Sundays out in shorts in the backyard reading JBC, and that's his favorite activity. But the advice he gave to me, and it wasn't exactly advice directed at me, he's kind of said it, and it's haunted me ever since. And he said, good ideas are a dime a dozen. It takes the real work to get on with it and prove it that makes a difference.
You're here. Thank you. Thank you very much, Scott. Okay, here we have.
Hi, my name is John Hentz. I teach Biology 107 here at WSU, and I went to the same college that Ralph Yount went to. I went to Wabash College in Crawfordsville, Indiana. Go Wabash, exactly. Yeah, so, oh yeah, any other Wabash grads? I would be surprised and impressed. So he got his degree from Wabash in 1954, and he, in 1999, was given the Distinguished Alumni Award. And in, what's that? Oh, and you went to that. Oh, yeah, nice. And in 2003, he was given an honorary doctorate from Wabash as well. And there have been a few times that I've been back to Wabash, and during my times there, very frequently someone from Wabash would ask me how Ralph was doing because they knew that I was coming back from Pullman. I grew up here, but I also have been here at WSU for 20 years. And they were always asking about Ralph because he was a huge contributor to Wabash as well. And Wabash was very important for him in his education and also just after that as well. So. Oh, he may have been the starting guard on football team. I don't know about his football history. I know he was a member of the Sphinx Club, which is a club there, sort of the Wabash's version of cheerleaders. It's an all-men's school, so you dig deep for your cheerleader squad, and I know that he was a big part of that as well.
All right, thanks a lot. Who else can we interest in? Oh, here we have one back here. Great.
Hi, my name is John Wyrick. I'm a faculty member here at WSU. And my connection to WSU was in part because of Ralph Yount. My dad and I, when I was 18 or 17 or something like that, came to visit WSU, and we also visited UW, and I grew up in Spokane, so we were trying to decide which college to go to, and we just were wandering around the halls and happened to run into Ralph Yount. And he talked to us about it and said, you should come here. Yeah, it's a great school, you get great education, and so I did. And then 3 of my siblings came too, so 4 of us all graduated from WSU from Ralph, and I'm back here as a faculty member. And, had a really great experience there with working with Keith Dunker and Yayu and a variety of other people at WSU. So I'm really thankful for Ralph. He was a great mentor and colleague and great presence at WSU. So, yeah.
Hi, my name is Dave Lawson. I was Ralph's last graduate student. Actually, I was the last graduate student to graduate. There was another Alex, who wasn't able to be here today, was the last graduate student to enter the lab. He got out before I did. People today have talked about Ralph's kindness and compassion, and I'm going to add patience to that and believing in you, because as my research went on 5, 6, 7, 8 years, he supported me and kept me in the lab and helped me find my way. And so for that, I'm very, very grateful, and taught me to do good science. And that's still trying to do that today in the pharmaceutical biotech industry. I have one quick story that I want to tell. And a couple of years ago, I came up to give a seminar, and I looked in the faculty list. The only person on faculty that was here when I was here was Mike Griswold. So I emailed him. I said, "Hey, Mike, can I invite myself to give a seminar?" And Mike said, "Yeah, sure. It's summer. Nobody's going to be there." But he said, "I'll make sure Ralph's there." So he helped Ralph come to the seminar. Ralph's sitting up front, and I'm speaking to a room over in the new building about this size, and Ralph goes, "Dave, you've got to speak up. You've got to project. You've got to speak to the back row of the audience." I know people here have heard that from him. I'm not the only one. Happened like 3 or 4 times. "Dave, speak up. You got to speak up." And I get through the seminar, and we start chatting afterwards. He goes, "Oh, Dave, I'm sorry. I figured out that my batteries in my hearing aid died." He then went on to tell me that I had too much information on my slides. So, once your graduate advisor, always your graduate advisor. So, fondly remember Ralph in my time here in his lab. So, thanks.
That's great. Oh, here we have another one, Todd.
So I'm Todd Garabedian. I was a graduate student in Ralph's lab from 1986 to 1991. And I remember one piece of advice that Ralph gave to all of his graduate students, male or female, is he always said, put your gonads in the freezer during graduate school. Well, I actually didn't take that piece of advice because in Fulmer Hall, we were on the 5th floor, and across the way was the membrane group with Jerry Hazelbauer and Lin Randall. And one of Lin's graduate students, Carol Teschke, and I got together, and we eventually got married here in Pullman, and then we moved away, but formed many, many relationships here. But I do have fond memories with Ralph, the rigorous exams and the rigorous penetrating questions that he would ask on your research, just sort of follow you. After graduate school, I took a bit of a different path. I went on to law school, and I'm now a patent attorney. I've been practicing patent law for about 30 years. But I have to say, I use my science every day, helping my clients. And in some cases, I have to design experiments for them to do. So all of that rigorous training really paid off, and Ralph was a big part of that.
Thank you, Todd. Some more folks. Here we go.
Just one more anecdote. My wife and I took a side trip when we were coming to Pullman to accept the job. I went to Dallas, Texas, and gave a seminar. And so we arrived in Pullman after our furniture and cars had been shipped. And of course, the shippers won't unload the things unless they're paid. And so Ralph, out of his pocket, paid $1,000 to get our furniture unloaded. I mean, that's just the kind of guy he was.
Amazing. Yeah.
Of course.
Oh, yeah. What do you feel comfortable with now? Yeah?
Yeah, I took
Great.
it out.
Yeah, okay.
Okay. Hi, I'm not a scientist. I'm his daughter. I also don't have my dad's extemporaneous comfort in front of a microphone, so I took some notes. But for the friends who haven't been able to see him for a while, I want you to know that the dining table looked like his office in Fulmer Hall. It was stacked with periodicals and papers and raspberry jam and chocolate. And he kind of had everything going on there. And the two things he kept saying over the past 20 years or so is, I can't believe I've lived this long. I can't believe I've lived this long. I'm so lucky. I'm a waste of resources. like, no, you're not. No, you're not. But I think, well, he was lucky because he loved his work. You guys are his family, really. And he also had fabulous neighbors like Dwight and Peggy Hagahara, and great helpers, caregivers like Winnie Harrington right here, who let him live in his own home that he built with my mom until he was 93, surrounded by trees that they planted. And so he was a really lucky guy. And what else did I want to say? Oh, when Jim had the slide that said Ralph to the rescue, I think Jim came to the rescue for us because our family was so small. And after he died, people said, you know, are you going to have a memorial service? And I was like, well, the only thing I saw was the Psalm of Myosin, which he had on the wall by his computer, and that's about as religious as he got. So I'm like, wow, what are we going to do? And family's small, and Jim's like, hey, let's get everybody together. And so this is perfect. He would have loved it, you know, science and education. So thanks.
All right. Thank you. Are there others that want to say a few words? Oh, okay. Ed.
I'm Ed Rubio, the son-in-law. Married Allison here. One of you mentioned fireside chats, and I feel like I had 30 years of fireside chats with Ralph. One of the photos here is his float home that he and Valerie purchased in 1998, and we often sat at the table, and usually the topics were three things: politics, that was mentioned tonight, science, of course, also his family. And very often, his family were his colleagues and his former students. Looking at the program tonight, I kind of got to thinking, I already kind of know some of the most prominent protein scientists in this country. And it's because of the times that Ralph often talked about all of you. I understood maybe 10% of the science he discussed, but what I truly understood was the passion that he expressed in referring to his colleagues and former students. Same kind of passion I saw tonight when you were talking about your science. And so I guess what I'd like to say is that he was so proud of all of you, and I think that's where he was coming from. That's it.
All right. Thank you. Okay, Yayu.
Well, I didn't hear anybody talk about his democratic activities, so I thought I'll supply that dimension. I was very active in the Democratic Party here, rose to the level of us being the vice chair of the state. But I have so many volunteers for me, but the most fervent volunteer was Ralph Yount. He didn't want to seek any political office or anything, but anytime I have a function, he's a supporter. Anytime I need people doorbelling, he went doorbelling for me, and for us, So, I mean, he was really, really a great Democrat. But when I was, when he became the president of Biophysics, I was the treasurer of the Chinese Biophysics Society because for years I supported it. So I say, our group's going to meet. Would you come and talk to us? In the Chinese Biophysics Society. And I thought he was so busy he wouldn't, consent to, but he did. And he came and talked to us, and it's the first time our Chinese Biophysics Society has a speaker. So I thought I would supply that.
Awesome. Thank you, Yayu. All right. Oh, more. Yeah. Michael.
My name is Mike Hintz. I worked in the storeroom at for chemistry and biochemistry. And you've already heard from my son, John, but my other son, Bill, also went to Wabash College. And he worked 2 of his summers while he was in college in Ralph's lab. And I was disappointed that Jean Grammer didn't come here because she was his mentor as far as he learned all his laboratory techniques from her. And Bill once told me that, he said, she knows everything. And every lab needs one of those people that knows everything.
Thank you, Michael.
By the way, Bill went on to become Dr. Bill, and he's a rheumatologist in Eugene, Oregon.
Oh, good. Yeah, thank you. Jean couldn't be here. I think she's migrated to New Orleans. She does so every 6 months. Are there any others that would like to say anything? Okay. Ross?
Yeah, this is one story where Ralph really got upset at me. We had gone to the Biophysical Society meeting, and it must have been 1979 or maybe 1980, and we took the Washington State University van to San Diego. And he learned, actually, it took 3 or 4 years after that when he learned that we had taken the Washington State van into Tijuana, Mexico. And he goes, Ralph, he goes, Ross, are you serious? You did? So anyhow, he really was quite upset at me, but I blamed it, of course, on my other 3 people that were in the van. So
That's okay. What? Okay. All right. Jerry?
Well, I was going to say this a little bit ago, but everything was so flowery. I thought it'd be good to point out a different side of Ralph. He was dishonest and fearful, the opposite of what we've heard. There was a tech that I mentioned by name earlier, and I'm going to forget that name now, but Ralph didn't like her. None of the graduate students liked her. Other tech didn't like her. And she constantly emptied Fulmer, I think it was dibromoacetone or something, it was vile. But she also failed to change out the reagents on the shelf, and nothing was labeled, nothing was dated, and people complained. So I was, being the youngest person, just an undergraduate, Ralph called me in his office. He said, I want you to come in on Sunday, and everything that everything that this person has that's not labeled properly, I'm talking about freezer and fridger also, I want it dumped. And I said, well, why don't you just tell her? Oh, no, I'm afraid of her. So anyway, I went in and did what he said, and I dumped out probably 50 bottles of reagents from the shelves, probably that many from the fridger, and a whole bunch of tests in the freezer that weren't properly labeled. And she came in Monday morning just absolutely furious. And I heard in her office screaming at Ralph, his office screaming at Ralph. I heard Ralph say, don't worry, I'll take care of it. He doesn't know any better. He thought he was doing right. So he said, I'll talk to him right now. And so he called me into his office, and he started raising his voice very loudly at me, and he was smiling the whole time. So he let me take the rap on that completely. Wasn't totally honest and wasn't totally fearless.
All right, thanks. Jerry, yeah. Okay, we want to get a group photo with all of us here. And so we're going to do it up here. And we'll have, I think what we want to do is let's have the recruited faculty sit on the ledge here, right here. Can you all come up? We're going to take a photograph from this side with all of us who attended. Is that okay? Raise your hand if you're okay with that. I'm seeing hands up. Okay, let's do it. Come on up here. Yeah, what do you think? We can do that? Yeah. Well, okay, that's good. All right, we can do that. Yeah. Okay, so Mike, I think you and I ought to be on either side of this podium here.
Pull them out so people can stand behind as well.
Oh, that's good. Yeah, that's a good idea. Yeah. Okay. Yeah. Okay. Yeah, have a seat.
Who's sitting on them?
You're sitting on it.
Okay.
Sit right there. Where is Allison and Ed? Could you guys come here? Yeah, we're going to have you guys right here. Allison, sit. Well, that's okay. I can have you here. And Ed, we'll have you there. Then other recruited faculty here. And then the students, the grad students, let's come behind them. No. Go ahead in there, Ross. Next to, come on in, Brian. I don't know you. Yeah, grad students behind these guys here. Over here. Yeah, Mickey? Yeah, yeah. Right there. Where's Gary? Okay, Gary's got to get in. Okay, so we can do a row up here too. So you guys, all you guys have to be in this. So come on up. You raised your hands. Okay. So come on up.
That's in front of podium here.
No, well, that's okay. Yeah. Mike and I can get in here. Okay, we can get in here. Hervé, you're slow to the party here, dude. Okay. Everyone has to be visible. Yeah. Hold on. Okay.
I'll stand here and you stand there.
All right.
And you'll be so pretty.
Okay. We all? Okay. Can everyone, so which cameras are going to be, I think there's 3 of them back there, right? Okay, so raise your hand when you're ready to go. And ask us to shout out Ralph. And we don't mean barf either. All right, tell us when. Okay, some other people in the back, like Gerhard Munsky, get over on the side over there. Okay.
Can't see the camera.
Yeah.
They can't see you.
Yeah. They can't see you. So Julie, you're going to have to move one side or the other.
Yeah.
Okay.
I'm fine.
To Ralph. Ralph. 3, 2, 1. Ralph. All right, thank you so much. And let's give an applause for these guys back here who did so much.